All right, thank you. Welcome to the Biological Tools Workshop. Thank you all for coming. I'd like to start by thanking our speakers, discussion leaders, my co-organizers, Luke Lavis and Zhuoran Ma, and especially Janine Stevens and her team at Janelia for helping us put this on. This is the fourth in a series of 10 workshops to help us shape a new 15-lab, 15-year research program at Janelia called 4D Cellular Physiology, or 4DCP. The goal is to understand the function, structure, and communication mechanisms of cells within and across organ systems. Our goal with this workshop was to invite people doing exciting cutting-edge research in imaging and technology development defined rather broadly, particularly with respect to what Janelia is known for, in the hopes of stimulating discussion and fostering new collaborations between the Janelia community and the community at large. I'm really excited to hear from our speakers over the next 2 days about their research and visions for their respective fields and for the discussions following their talks. So I'll hand it over to Luke now.
All right, so I'll just echo Alison and thank the speakers, the discussion leaders, and all the attendees, along with Janine and Letha and our amazing AV team. Thank you for participating in this workshop. I'm kind of the old timer at Janelia now, and I've lived and breathed this crazy collaborative environment where biologists and tool builders work together to solve important problems. And as Alison mentioned, this new research area is rather broad. I like to refer to it as not plants. And so it will require investments in new technologies and collaborations with new tool builders. And like Alison, I'm really excited to hear talks from this variety of scientists, frankly, most of whom I've never heard speak. So again, thank you, and I'll turn it over to our co-organizer, Zhuoran Ma from NCI.
So, hi, my name is Zhuoran Ma. I'm a PI at the NCI and the shadow third organizer. So, you know, when Luke and Alison asked me to help out with this conference, I gotta say, you know, I really wasn't kind of sure what to expect. And in some ways, I think, you know, the most exciting thing about this conference is that I still don't know what to expect. I think there's going to be such a vast sort of array of different tools that are going to be covered. And I would say every organizer, you know, has, you know, some intellectual, you know, tendrils that reach some of these fields, but none have all of them. And so I think that's the kind of conference that gets me really excited because you can really learn something, right? Not just talk to your friends. So yeah, I want to thank, of course, the organizers, you know, Luke, Alison, and Janine, and HHMI, but then really just all of you for kind of taking the leap and trying this. It's kind of a new kind of conference. I think it's going to be really enjoyable and exciting. So thank you. I'll turn it over to Janine, I think, for a few logistical comments.
Yes. Hi, everyone. Just some quick notes about the format of the workshop. All of our talks are 10 minutes long, and the speakers have promised me that they will not go over, at least not by much. So we will have a solid 4 or 5 minutes after each talk for dedicated audience questions. So please write your questions in the Q&A box at any time, you know, as they come to you. And once the talk is over, the chair will go ahead and read them out loud to the speakers. 5 minutes is not a lot of time, but we will try to get to as many as we can. There's also a 20-minute discussion period at the end of each session. This time is not necessarily intended for specific questions for each speaker, but really more for higher level and forward-looking conversation about the tools and technologies that we've just heard about. But again, we still encourage you to share your questions and any thoughts you have in the Q&A box and the chat box during the discussions. Just as a reminder, all of these sessions are being recorded and they will be available on the workshop website at janelia.org within the next couple of days. And with that, I'm going to turn it back over to Alison Tebo, who is chairing the first session. Alison.
Great, so we have an exciting session on imaging very broadly, and Bernd Bodenmiller from ETH Zurich is going to start us off.
Okay, thanks for the nice introduction. I hope I will be able to use the 10 minutes productively. So my group is interested in developing tools to better understand tumor ecosystems in the context of precision medicine applications. And if you think about a tumor, then it contains many different subtypes of tumor cells These are embedded in the stroma. There are other normal cells like immune cells, fibroblasts, nerve cells, etc. And all of these function and act together and communicate to form a dynamic ecosystem. Now, if we want to study a tumor ecosystem, we want to accurately know which are the cells that are present in a tumor. We want to know the overall composition of a tumor. We want to know where are the cells located, how do they communicate with each other, what molecules are used for that. We want to know for every cell which pathways are activated, deregulated. We also want to know the overall 3D morphology. And the only way how we can do that is to do multi-scale measurements from the level of molecules all the way to the 3D morphology of a tumor. And we would like to know this because each of these scales, from the cells, from the pathways to 3D morphology, to the cell-to-cell interactions, are very potent targets for drugs in precision medicine applications. In addition, having measurements of a tumor across all of these scales is also highly informative for cancer biology in general, which always is the core for best patient stratification and identification of better treatments of patients. So my group developed to measure tumor ecosystems what is now known as imaging mass cytometry. And here we take routine tissue sections, we stain those with antibodies that carry pure metal isotopes, we scan with a laser over the tissue that removes tiny particles of the tissue with subcellular resolution. We take a mass cytometer to read out the metal content of each location where we shot with the laser. And then we can in silico reassemble an image of the tissue with subcellular resolution. And by now we can routinely image over 40 markers simultaneously in situ with imaging mass cytometry. Why metals over fluorophores? Well, fluorophores have many issues. They overlap in their emission spectra, also excitation is difficult. And by using metal isotopes, we pretty much solve all of these issues and some others that you will see in a couple of slides. Now, here's an example of a cell line that we imaged with imaging mass cytometry. And I love to show you especially phosphorylation on histone H3, which is a mitotic mark. And you can see here how the chromosomes are condensed, they are pulled apart into the daughter cells. Here's another example of an MDA-MB231 breast cancer cell line you see in green vimentin. And now zooming into those images, you can nicely see even vimentin filaments. Over here, you see another cell in mitosis. And what we have learned now, since we developed this technology, is that the data can be very well compared to routine fluorescence microscopy, but we have more than 40 markers measured compared to fluorescence microscopy. So now there are also some advantages to fluorescence-based microscopy. To the left, you see a bone metastasis of a breast cancer patient. I zoom into a region here, and now you can see this tissue measured with fluorescence microscopy. Lots of yellow that indicates pancytokeratin. But now if you look at the same region in the imaging mass cytometry, you do not see any signal. And the reason is that especially formalin-fixed paraffin-embedded tissues, they strongly suffer from autofluorescence, and we do not deal with that. We also overcome other issues such as tiling effects in fluorescence imaging, or in case people do serial antibody stainings to do multiplexing, then there's an issue of optimizing the antibody order. And we have a dynamic range that's 100 to 1,000 times higher to fluorescent-based approaches. We also developed tools that enable us to simultaneously measure transcripts in situ with high sensitivity. We have a detection a limit of about 10 copies per cell, where we then simultaneously, together with the transcripts, again measure the antibodies. And in a moment, we can simultaneously measure 12 transcripts with 28 proteins, but the number of transcripts we can measure is continuously increasing. And this is just a zoom in, where we again see different phenotypic markers, but then in italics, you see transcripts that we measured here in this breast tumor section. And this one, I think, was like 3 × 4 millimeters that we had actually analyzed. So then I talked before about the 3-dimensional analysis of tissues. We are also developing methods on how to enable 3D imaging mass cytometry. So there are 2 approaches that we developed, and now is fairly mature, and the manuscript was just recently resent for review. We take here tissue block, we do serial sectioning using a customized diamond knife setup. We then do the antibody staining with the metal antibodies of these many sections. We do the imaging, we do image registration. We build a 3D voxel model where now each voxel contains, again, information on 40 different antibodies. We do 3D segmentation, and then we visualize the images and volumes. And by now we can really generate, I think, quite detailed 3D models of tissues. Here you see a HER2 positive breast cancer tissue. You can see in yellow cytokeratin, so this demarcates tumor regions. You see in red, that's a marker for blood vessels, and in white, you see actually the immune cells being in the stroma outside of the tumor region. Now, after 3D segmentation, we get tumor models akin to this one, where you see in orange the individual tumor cells, and then in between, again, based on vimentin and smooth methyl actin, we can see immune and stromal cells within the 3D structure of the tumor. Now, why 3D? People always ask me, well, my answer is we live in a 3D world, but not everyone's convinced. But to convince you why 3D is useful compared to 2D, when we analyzed this 3D tumor model, we found one region that is shown up here in the circle and over here, where we found individual tumor cells. There's the tumor mass, then that's over here. Then we have a micro protrusion. And going away from this micro protrusion, we have individual tumor cells. And what we likely see, or what we can hypothesize, is that we see here a pseudo-spatio-temporal process, namely a tumor region that becomes invasive. We have the micro protrusion and the individual cells are coming out. Of course, it's a snapshot. That's why we can't say that this is actually happening. But to see such a process in situ, just based on a 2D section, is not something that you can really expect to see. Now, finally, we are also develop computational tools such as HistoCat or CytoMapper, where we can segment those images. We can determine the cell phenotypes. We developed a couple of algorithms that allow you to determine whether cells are more frequently next to each other than you would expect by chance or not. We have just published recently a paper in Nature where we show that cells can form communities, and that is highly informative in regards to the prognosis of patients. And there are also many other spatial analyses that you can do once you have masked the images or the 3D model. We can do the same there. And for example, ask what's the distance of immune cell to the tumor mass, etc. And all of this is freely available. You just have to go to our GitHub page. Just now here to summarize, then I'm done. The dynamic range of imaging mass cytometry is about 10 to the 5. We can look at any tissue as long as antibodies bind. Limit of detection similar to fluorescence microscopy. In the moment, it's still a slow technique. It takes about 1 hour to analyze a square millimeter, but it will become faster. The resolution is not as high, but close to fluorescence microscopy. We can measure transcripts, proteins, and modifications at the same time. We can do whole slide analysis. An important thing is we also developed protocols for high throughput screening of organoids and other 3D tissues. It was just published recently. And the technology is really, really robust and reproducible. We are part of a clinical study where this is continuously assessed. And I think that's a big strength of the technology. And by now, really, in the projects of my group, it's routine to look at thousands of patient samples within a project to do this multiplexed imaging. And recently, a postdoc of mine told me he just did 8,000 images in a pilot project, and I have to admit, I nearly fell off the chair. So with this, I greatly want to acknowledge my lab. I want to acknowledge these different institutions for providing tissue samples to me. And I also want to greatly acknowledge the funding agencies of my group, especially the Swiss National Foundation, the ERC, NIH, Cancer Research UK, and the HubMap program. And with this, I'm done, and I hope this was still within the 10 minutes. I'm happy to receive your questions.
Great, thanks, Bernd. That was a really great talk, and we have several questions. Just to one of my questions, just to get us started, is you mentioned you can look at up to 40 different targets. Is the main limit to that sort of number 40, is that have to do with the metal barcoding, or does it have to do with sort of like the number of really good antibodies you have that are interesting for your samples?
So it's not the number of existing antibodies. In our fridge, there are hundreds of them. The challenge in the moment is to identify chemistries that bind metal isotopes strong enough to antibodies. And in the moment, we mainly use lanthanide isotopes. They have a very similar chemistry. And so we can use the same polymer with the same chelator for all of these isotopes, and it's easy to label the antibodies. There's some elements like tin has 7 isotopes that we could access, but they really love to switch charge state. They're very difficult to handle. And so they're very difficult to bind onto the antibodies. And that's really the main challenge that we are faced with, is to find good chelators for metal binding.
Okay, great. So we had another question about what the main limitation for achieving higher resolution is.
So there are two. So just based on the technology, we already could scan with higher resolution. The downside, though, then is sensitivity, because what we have to balance is, for us, the number of molecules that we measure is, of course, proportional to the amount of tissue that we introduce into the machine. And so that's why we eventually settled at 1 micrometer, because that's really the sweet spot between, in the moment, throughput, sensitivity, and a sufficient resolution to address many of the questions that we're interested in. But you could do higher resolved imaging. We believe to have reagents that allow us to put more metals onto the antibodies. So this will then allow us to do higher resolved measurements at the same sensitivity. And ultimately, it's also a question of scanning time, because we do pixel by pixel by pixel. And so if you reduce your pixel size, you also increase your measurement time. And then these are just like the factors we need to balance in deciding where to dial in. And for our questions, the 1 micrometer is the sweet spot, as I mentioned. But for other questions, you might dial in even at the low resolution, and you get the answers that you actually want.
Could you
to that?
Oh yeah, go ahead, Kaspar.
Bernd, have you quantified the sensitivity in terms of number of molecules?
So we think that the sensitivity is in the range of low number of few hundreds of copies per pixel. That's the roughly where we are. So it's not single molecule detection. Well, there might be some exceptions when we can put some metal nanoparticles on antibodies, but there are only a few where this works. But I think realistically, it's like 100, 200, 300 copies per pixel. But in addition, it's also a function of the antibody. Because, you know, some antibodies, they don't bind really well. It's also a part is accessibility of the epitope. I mean, but these are just all the issues, not of the technology, but of using antibodies. And that's just an additional layer.
So if you could replace antibodies with something else, like what would be on your tick list of, properties of those binders?
That's a great question. If there was something better than antibodies, I think we had switched already. I mean, there are many properties. Well, I have to say, if I could choose, then really good antibody. All antibodies should be like really good antibodies, and I feel companies can produce really good antibodies, but rather often companies just throw onto the market and not validated antibodies, and it's up to the scientists to figure out whether they work or not. So I think my answer would be actually that companies really do their homework before bringing an antibody onto the market, and that would already solve lots of our issues.
Great. Well, that's all the time that we have for questions right now. I look forward to picking up some of these threads in the discussion later. So next up is Lixue from Caltech.
Is this the right screen?
Lou, we can actually see all of your slides. I think we're looking at a presenter view. There you go.
All right, so, all right, I'm ready to start. Hi, everyone, good morning. So I want to take this 10 minutes to share with you our recent efforts on pushing the free front for stimulate-Raman imaging for complex bioanalysis. So the stimulate-Raman SRS microscopy is a nonlinear Raman imaging modality that utilizes two pulsed laser, one for excitation and one for stimulation. When the energy difference between the two lasers matches with the vibrational motion of the chemical bond, this chemical bond this bond vibration of the interest would be get excited, that resulting in the loss of a pump photon and the gain of the Stokes photon. So we call this stimulate-Raman loss or stimulate-Raman gain. By filtering out the other laser and detecting either one of them with a large area photodiode, and by raster scan the laser spot across a sample, you can generate a SRS chemical map, that is the image. And by using a high frequency modulation transfer scheme, we can push the detection to a short noise limited region for high detection sensitivity. So although the SR spectroscopy was observed 50 years ago, it was not. It was demonstrated as a microscopy technique about 14 years ago, reported by a few groups. So it's, since the invention of SRs, it has been a very useful and widely popular label-free imaging scheme for which you can target the vibrational bond from an endogenous molecule and map out the distribution in, for example, for the total proteins or lipids in live cells and tissues. So one of the efforts in our lab is trying to develop a super resolution SRS imaging modality. While those efforts are ongoing, we have recently inspired by the success of a sample expansion strategy, identified and adopted a sample hydrogel hybridization strategy that would allow us to perform isotropic sample expansion without use of protease digestion, so that we can retain all the proteins and target a methyl group vibration to map out the protein-rich structures in tissues without, in cells and tissues without any labels. So this would allow us to push our resolution from the normal 350 nanometer to down to 91 nanometer in cells and also in tissues for high precision imaging. We termed our technique VISTA, and that would allow us to image the neuronal cell bodies and the processes in tissues in 3D. So without any labels, we also want to achieve imaging specificity. Motivated by that, we trained a machine learning algorithm and using fluorescence labeling as our ground truth for prediction, and train our VISTA images for predicted targets for on the VISTA sample image. So by that, we can achieve multiplex label-free imaging for nucleus, blood vessels, neuronal cells, and tissues with specificity close to that shown by the fluorescence labeling for antibodies across 3D in tissues. So I'm here to share you with my visions. So so I'm going to tell you some of the issues. So the issues with the label-free imaging is we sometimes lack specificity. That's why I've been using machine learning to gain specificity. So imaging with without any labels is great because it allows you to bypass all the labeling related issues. So for example, as in fluorescence, we don't have any photo pollution issues for fluorophores, and we don't need to worry about the inhomogeneous probe delivery into the deep tissues. But imaging with probes would give you much higher specificity and sensitivity. So by introducing a small vibrational tag, that is in essence just a small chemical bond, this minimal labeling strategy would allow SRS to go toward more functional imaging side, especially very desirable for imaging small molecules like small metabolites. With this very distinct Raman groups that gives you good Raman specificity. So with alkyne and carbon deuterium probes, we and others have demonstrated the imaging of broad spectrum of small metabolites from nucleic acids, amino acids, glucose, choline, lipids, and even deuterium oxide. For probing their metabolic uptake, synthesis, conversion, degradation, part of the dynamics in live cells and tissues, and even up to animals. So compared to mass spec-based imaging, SRS would provide you chemical specificity and the ability to track metabolites in live biological systems over time with high resolution. So with all of this established, both the label-free structural structural imaging chemical quantification and the labeled metabolic dynamic tracking, we recently trying to push this as a Raman-guided subcellular pharmacometabolic platform that would allow us to profile the metabolic processes in cancer systems. So we, in collaboration with Jim Heath, we applied this technique platform to probe the metastatic melanoma cells. So in short, on the single cell level, we were able to generate good correlations from our Raman spectroscopy and imaging measurement on live cells to the gene expression profiles on the single cell level. And on the subcellular level, we were able to identify a unique metabolic susceptibilities for the lipid monounsaturation. So by inhibiting the monounsaturation from one of the drug-resistant phenotypes, we observed a formation of a separated membrane domain inside cells, and with a correlation with LC mass and lipidomics, and also biochemical assays, we were able to basically figure out the potential metabolic control pictures underlying this metabolic susceptibility of monounsaturation toward this drug-resistant phenotype. I'm omitting a lot of details because I want to give you an overview of what SRS can do. But to be honest, the issue here is the sensitivity. So although we have live cell compatibility, we have minimum label, minimum perturbation, current barrier for further pushing the application into complex biological questions is the sensitivity. Currently, we're limited to about 100 micromolar of sensitivity for detection sensitivity for the small metabolites. So that is one of the fronts that we are actively pushing, trying to achieve higher sensitivity, ultimately, hopefully, toward single molecule detection. So in seeking high sensitivity, we also previously developed an electronic per-resonance SRS imaging modality. So the rationale behind it is that if you bring your laser close to the absorption peak of the molecule, but not directly exciting them, then the Raman signal would benefit from the huge resonance effect that would allow us to do the detection on a nanomolar region. So although now we're dealing with absorbing molecules because our laser is in the near infrared region, the molecule that works really well under this electronic resonance region is about the 700 nanometer absorbing molecules. So So with this nanomolar sensitivity, we were able to perform basically imaging with quality very close to fluorescence, detection limit very close to confocal fluorescence. And the added bonus is that the Raman spectrum is very narrow. It's about 100 times narrow compared to the absorption emission spectrum of fluorophore. So we get very high chemical selectivity. Here, this is targeting the double bond vibration of adenosine 740 molecule immunolabeled tubulin, and by just tuning the laser across away for 2 nanometers, you see the signal is gone. Since the broad absorption emission spectrum for fluorophore is what limits the multiplex, basically the multiplex barrier for fluorescence imaging, we envisioned this APRSS technique would ultimately allow up to 100 color imaging, with the prerequisites that we have to develop the matching Raman dyes for the applications. So this was a panel that we designed and synthesized before with a triple bond conjugated to a conjugation system of the absorbing molecule that would allow us to fine-tune and achieve this fine-spaced peaks across the Raman spectrum. And so far we have about reported about the 16 color imaging, but on tissues we demonstrate 8 color imagings. While we're still trying to keep increasing the number of the Raman dyes that we could develop an image for multiplex purposes. One of the efforts we're also trying to push is the develop photo switchable pre-resonance probes by modulating the coupling of electronic and vibrational coupling for the probe, so that we could modulate the electronic states to achieve the reversibly switchable SRS probes for the signals toward a super multiplex super resolution imaging. So with that, I'm over my time limit a little bit, and I'd like to acknowledge my wonderful group and also the funding agency. Thanks a lot.
Great. Thanks, Lou. That was a really great talk. So one thing I was a little bit curious about listening to your talk was you said that it was live cell compatible. What's the time resolution for this type of microscopy? Is it significantly different from fluorescence microscopy, or around the same?
Right, that's a good question. Thanks, Alison. So it depends on the signal size. Because there's a signal to noise ratio limited. On the faster side, I would say on the typical side, is very close to the frame rate, the time resolution for fluorescence. With higher level of signals, we can go up to a few frames per second. With a typical rate is about a few seconds per frame. So for all of those alkyne and carbon deuterium labeled molecules, it's about somewhere between 1 to 10 seconds, depends on our signal noise ratio.
Okay, that makes sense. Kaspar, did you have a question?
Yeah, I have a few questions, Lou. We'll see how many we get through. So, you mentioned that there's no bleaching, right? And so I guess I think a lot of people want to know what limits the sensitivity, at least, where we don't care that much about frame rate. Could we integrate for an infinite amount of time and get much more sensitivity?
That is a good question. I think the answer is yes. You can keep averaging them as long as so there's a trade-off, right? So for us, if we want to perform live cell imaging, you don't want to keep averaging forever to minimize any phototoxicity. But to increase the sensitivity, you can definitely just doing much longer precision. For example, for now, we do a pixel dual of about tenths of microseconds. So that gives us a few seconds per frame. We can definitely do a millisecond per pixel. Then that gives us maybe a minute, a couple of minutes a frame. Then that brings the sensitivity down to a few fold or tenfold to increase the sensitivity. Yeah, we can definitely do that.
I have another question for sort of tissue imaging. I know that this requires sort of, or maybe not doesn't require, but usually it's a forward scattering method. And so how much worse is it if we have to image in a thick sample where we only have access to the top, for example?
Yeah, that is a great question. So, well, if you think about the technique, it's actually quite similar to two-photon fluorescence. It's both of a near-infrared laser, two-photon sectioning. So the tissue penetration, as we go in, we could image up to a few hundred micrometer without any tissue treatment. Of course, we can adopt the tissue treatment for tissue clearing or expansion base that would allow us to image much deeper, a few millimeters. So right now on the untreated tissue, we can image down to a few micrometer. We haven't really quantified what kind of loss of signal do we have that we have as we go inside the tissue. So it's more scattering. But I think it's both of a loss of a signal noise ratio, lower of a signal noise ratio, and also the increase of signal to background ratio, because we start to generate a lot of background signals as we go in. Yeah, I don't have a quantification metric for that, but we can. Depending on the scattering parameters, we can image anywhere between 100 micron to a few hundred micron inside tissues.
Great. So that wraps up our time for questions. Thank you again, Lou. Our next speaker is Lixue Shi from Columbia.
Thanks, Alison, for the introduction. Also, thanks to the organizers for this great opportunity to be here and introduce you how chemical imaging contributes to understanding metabolism. Okay, studying metabolism is indispensable to unraveling the mechanism of many biological processes, such as development, aging, and homeostasis. Although the importance of metabolism is clear, two developments to obtain in situ metabolic activities, especially with subcellular resolution, is relatively lagging behind. Clinical widely used PET and MRI usually lack sufficient spatial resolution to resolve single cells. Fluorescence microscope can visualize metabolism with subcellular resolution, but requires bulky fluorescent labeling, which is perturbative to normal biological functions. Mass spec imaging requires a long analysis time, especially with fine spatial resolution. As Lou has just introduced, Raman microscope has a major exciting advance in past several years, utilizing vibrational probes for tracking small molecules. However, the throughput is still relatively low, largely due to the notoriously weak scattering effect. Complementary to Raman scattering, IR absorption is an alternative chemical contrast which has a significantly larger cross-section. Its absorption cross-section is roughly 10 orders of magnitude stronger than the Raman counterpart. Besides these large cross-sections, instrument-wise advances in both the light source and the detector made mid-IR imaging a high throughput tool for molecular analysis. FTIR microscope has now been commercialized with a large area focal plane array detector for biomedical imaging purpose. New light sources, including synchrotron and quantum cascade laser, provide much stronger power intensity. In particular, quantum cascade laser enables narrow-band tunable excitation and renders a new framework of discrete frequency mid-IR imaging. While ion microspectroscopy has run for decades and widely adopted in fundamental research, the detection it is always operated under the label-free diagram and does not use probes. In this regard, the detection specificity is usually compromised and the information of dynamic metabolism is in lag. So to transform that, our work introduced a set of small vibrational probes designed to profile metabolism in cells and animal. We designed 3 categories of probes, including azide, carbon-13 substitution, and carbon deuterium, each with a different focus. The first group is the azide. The large molar extinction coefficient confers azide a desirable probe with a single, narrow, and strong band in the cell-silent spectral window, where no endogenous biomolecules vibrate. Therefore, azide is very useful to trace a single type of amino acids or fatty acids. For example, we can image the methionine analog AHA in cells or azide-labeled palmitic acid in C. elegans. The second group is carbon-13 substituted metabolic precursors, which will shift the amide or the ester carbonium bands by 30 to 40 with number. Then the carbon flow of during the biomass production, like protein synthesis, can be tracked by the ratio of carbon-13 substituted band over a regular band. Finally, carbon deuterium probe is a very successful tag in Raman, which is also IR active. Though the extinction coefficient is low, CD probe advanced in good labeling availability, especially for animal experiments. So for instance, by administrating D7 glucose in lactating mice drinking water, glucose anabolism of the pulp mouse during development can then be detected on brain. So with this entire library of IR active metabolic precursors we have built, below I will give two examples to show how they can be utilized. For metabolism mediated probes, the targeted IR modes only appear after metabolism of the labeled precursors. Therefore, they can be applied for monitoring a particular biosynthetic pathways. For example, deuterium water can participate in synthesis of a variety of CD containing macromolecules. So by feeding mice with D2O in their drinking water, we can monitor CD signal accumulation as a broad peak appears in the spectral range of 2100 to 2200 with number. Interestingly, two distinct peaks were observed, but with quite a different spatial distribution. So based on our in vitro spectral analysis, these two peaks are indeed attributed to new lipids and new proteins. Therefore, protein and lipid synthesis rates can be visualized simultaneously in a quantitative way based on true ratio maps of CD protein over amide peak and CD lipid over CH lipid. The other example is that these probes allow multiplex metabolic imaging. Here, by adding three types of probes together into the culture medium of adipocytes, we demonstrated multicolor imaging on protein synthesis, lipid uptake, and glucose anabolism together. So with this successful coupling of vibrational probes to immunoIR microscopes, our technique is uniquely suited to metabolic imaging with high information throughput. To showcase that, we performed two types of applications. The first one is for single-cell metabolic profiling. So to perform a metabolic activity-based cell type classification, we chose two probes, azidopalmitic acid and deuterium-labeled amino acids, to monitor lipid uptake and protein synthesis, which are two most important metrics in metabolism. Remarkably, three different breast cancer subtypes can be well classified readily on a 2D scatter plot based on lipid uptake and protein synthesis rates. In addition, the order of the metabolic activity agrees well with the characterized aggressiveness of these three subtypes. Besides cell subtyping, cellular heterogeneity can also be studied on a large cell population with the unprecedented speed of discrete frequency IR. Here, human breast cancer cells were exposed to FDA-approved chemotherapeutic drug Taxol in together with carbon-13 amino acids labeling. The protein turnover rate of over 15,000 cells were measured in only 3 to 5 minutes, showing the incredible speed of middle-IR metabolic profiling. Also, evaluated by the coefficient of variation, we found cellular heterogeneity is significantly elevated under the Taxol treatment. Such a small effect is easily missed without this high throughput capability. The second application is for large area metabolic imaging. For example, we generated a tissue atlas of micromolecules synthesis in the D2O labeled mice. In addition, we also studied the metabolic activity in glioblastoma multiform mouse model during tumor progression. Highly activities is observed inside tumor compared to this surrounding normal tissues. Also, our results suggest the existence of a metabolic active infiltrating front, especially in this late stage of tumor growth. So to summarize, we introduced a IR imaging-based framework of high-content, high-throughput metabolic imaging enabled by versatile small IR tags. Our technique is suitable for studies in which fast speed or rich spectral information is essential, while as the submicron resolution is not necessary. With that, we have demonstrated the applicability in single-cell metabolic profiling and also tissue-to-organ level metabolic imaging. For the future, I envision the rich information obtained from mid-IR metabolic imaging is particularly powerful for data-driven biological discoveries. For example, to study the intratumor heterogeneity in situ and decipher local communities with both chemical and activity information. Technically, the idea of vibrational probes is a general concept for all IR absorption-based imaging contrast. Further technical improvements reside in several fronts under our 4D imaging context. For example, how to improve the resolution and further extend the technique to 3 dimension could be addressed with emerging mid-IR imaging techniques, use new detection schemes such as photothermal and optoacoustic. In addition, life cell comparability and in vivo applications in large animals could also be promising with these new platforms, as they are much less sensitive to the water background. Finally, correlating metabolic activities with other dimensions, such as proteome, transcriptome, and proteome, could generate more insights for physiology and pathology. In the end, I want to thank my advisor, Professor Wei Ming, for his guidance and advice, my collaborators in Columbia and Data Solutions. Also, many thanks to the NIH to support this work. Thank you all for your kind attention. I'm happy to take any questions you may have.
Great, thank you, Lixue, for that wonderful talk. It was super exciting. I was wondering if you could talk a little bit about, further perspectives for multiplexing. So you showed us three different types of IR active probes. Where do you think probe design should go, and what do you think the limit there would be in terms of being able to really fully utilize that window?
Yeah, so based on our knowledge or our study with the Raman probes, as we have just showed, they're mostly relied on the triple bonds like alkyne and nitrile, which can be easily extended to multicolors with a long conjugated system or with the isotope substitution. But for IR probes, currently I don't have a clear vision on how to develop those multiplex probes, because one of the most stronger or interested target is AZ. For AZ, it is very hard to play with and to do the isotope substitution, and there is also a very limited color can be generated from this probe. So if we can have a module that is IR active, which can be tuned chemically to get more probes, that I think the multicolor potential is definitely very promising.
Great, thanks.
One of your directions, Lixue, was towards chemically specific interactions. Can you expand on that more? Is that just a matter of getting more colors, or is it a matter of targeting these probes?
Yeah, I think for one of the point we hope to use IR techniques is because they have a broad spectral coverage. We can have the information from our probe as well as all endogenous biomolecules. That's the chemical information we can extract directly. So if we can integrate the activity information we get from the metabolic probes with this intrinsic biomolecules or these intrinsic chemical bonds, I think that will definitely offer more insights or more correlations on what's happening in this local environment.
Luke, did you have a question?
Yeah, that was a really wonderful talk. I guess I usually think that we can buy a lot of isotopically labeled metabolites, but is that really the case? Or, are the commercial sources for the things that you want to do sufficient? Or, do you need other things with deuterium or carbon-13 or others?
Yeah, I think because there is a big field of NMR studies, so carbon-13 substitution and the deuterium substitution is very well developed for some like commercial sources. So I think for these two types of isotopes, they are currently okay. Yeah, but for other things like AZ, it is definitely, I think it's definitely underexplored.
So building off of that, could you use like azide tagged neurotransmitters to monitor like their packaging? Do you have enough resolution for that? Have you thought about doing anything like that?
I think first, our current resolution is about a few micron because we are using very long wavelength excitation, and also for this kind of wavelengths, it is very hard to get very high NA objective. So, but there's many other new techniques, as I showed at last. For example, the photothermal detection, because they use a visible light to excite, so the resolution can go to submicron, and they can do they have good compatibility with live samples. So I think if the sensitivity is enough, it's definitely worth trying.
Laura? Oh, sorry.
This is just maybe an off-the-wall question, but I wonder, people tend to use the same functional groups, and have you thought about using things like oximes or nitro compounds, because those seem like they would be really IR active and out of the spectral range and maybe not that hard to make?
Yeah, that's a very good point. We have tried a lot. Yeah, actually, we have explored a lot on the nitro group, but it seems this group is very environmental sensitive, not only for the frequency but for the intensity. So it's based on our experience on these commercial compounds. It seems the intensity will be influenced a little bit depends on like what the environment this probe will be. So I think if we can have more like input from the chemical side, I mean, if we have, because they are not designed for IR probes, so we can always try commercial things. So if there is like specially designed probes with these moieties, I think that's definitely very exciting.
Great, thanks, Lixue. So now we're heading into a discussion session with our speakers, and then also my co-discussion leader, Kaspar, who's going to help moderate and hopefully stimulate discussion.
Hey, I wanted to let you know, Alison and Kaspar, that Bernd had to log off. Hopefully he will be able to join us again, but you can go ahead and start the discussion without him.
Okay. Kaspar, you want to kick us off?
Sure. There was a great question for Lou that we didn't get to because it came right at the end about whether fluorescent proteins could be tuned with this resonant absorption-based method in the same way as the dyes. And I think that we could talk about that kind of thing more generally in terms of genetic targeting of some of these IR methods and how we could combine some of the expertise that that Janelia has in protein engineering and maybe combining proteins and chemicals with some of the things that you talked about today.
Right. So yes, the answer is definitely yes. So toward the end, I also actually showed the slides that we're trying to do reversibly switchable Raman probes as a resonance Raman probes. So that is actually the data shown on a bacterial phytochrome fluorescent proteins. So because of their absorption in the switchable in the absorption spectrum, we were able to achieve a reversible switchable Raman peaks from their double bond mode. So that is a great question. The challenge here is we can definitely, the answer is definitely yes, we can do the detection. But if we want to push toward the multiplex detection, then we would need resolvable peaks. So that's why we like carbon deuterium, alkyne, and Lixue like AZ so much, is because they're peak in the cell silent region. And especially for alkynes, a single very large Raman peaks that gives us a lot of ways to modulate its frequency so that we can, using isotope labeling, changing of the resonance structure to shift its peaks. So I would say the challenge, if just on the purpose of multiplex imaging, is how we can engineer the fluorescent proteins and then tune their vibrational frequencies away from each other.
I mean, or we could capture dyes that are tagged with proteins and maybe avoid the need to tune the proteins. Yes,
that's something we're actually being actively thinking about, how to make use of the great fluorescent proteins.
Can you imagine, Lixue, something similar for IR imaging and metabolic imaging?
I think for IR probe development, like the future is more elusive and require more imagination. So if we want to do genetic encoded, I think the purpose is to image a specific protein or other interested biomolecules with higher specificity. With that, I think the first thing we probably need to think about is the resolution and the sensitivity. For regular direct absorption detection, we definitely don't have the resolution to capture that information. You probably don't want to know the entire information of a cell in terms of our specific protein. And for the sensitivity, as I have a lot of other detection schemes, so if that really depends on like what's your particular question. So if we hope to really look at our specific protein with high multiplexing, I do prefer Raman actually at current stage. Because there is no necessity to really make IR do this thing. That's my feeling. I think we probably need to find the good technique for our question rather than make the question suitable for make the technique suitable.
So kind of building off of that last thought, what do you think some of the barriers to widespread adoption of these types of microscopies are for biologists or for end users?
So one motivation for us to pursue IR is because a lot of IR instruments have already been commercialized. All what I have showed in our work, we are using commercial systems. So we think that will get off the barrier for real biological or biomedical researchers to use these techniques. So we think our techniques is more suitable for some studies that you require a large data set, rich information, and very on a large area or a lot of cells. And it's also more accessible because for coherent Raman, you probably at current stage you still need to have some knowledge on this. But for FTIR, it's already very mature and you can directly purchase it.
Lou, do you have any thoughts?
Yes, I actually have a lot of thoughts on that.
Great.
So I totally agree with Lixue. So I guess from one front is on the availability of the instruments. I mean, if there's a commercial, just off the shelf Stephen Raman microscope that could be purchased, I guess that would allow the more wider availability, accessibility. But it's actually not that it takes some, I guess, inner knowledge, but it's actually not that complicated to build. So I guess in Janelia, we definitely have the expertise to build one. So yeah, I guess if we could push the commercialization on the instrumentation, that would be great. On the second one is actually following what Luke's question before on the commercial availability of the probes. I think for the one of the bottleneck is actually is availability of those probes. While the isotope labeled molecules are quite widely available, we can actually get them from purchase them, but they're very expensive. Mostly because we need, for example, if we need to culture the cells with the deuterated medium, we basically need to buy each of the components and then make custom made our own medium based on the formalism of the cell culture medium, and that same applies to the tissue culture and also for feeding the mice. So if similar, I guess, already made medium could become commercially available, that could also increase much, I would say, lower the barrier a lot in terms of biological applications and also the price. The third one, third limit, I think, is on the, as I said throughout my talk, is still on the limit, on the limitation of the sensitivity that we are striking to increase the sensitivity now. But I also think there's no single technique that could do it all, right? So there's always a niche for certain techniques. And I think the niche for StimulRama is we have very high biocompatibility. We can offer dynamical information in live cells and tissues without large labeling as required for fluorescence or for mass spec. We have live cell compatibility, no sample destruction, subcellular resolution. So those are, I would say, the niche for StimulRama. We just need a question that I guess I would say probably to stimulate more thinkings along the line that in which we don't actually need single molecule sensitivity, but we need more of a non-invasive dynamic applications. I guess that's the question I'm also actively thinking about. And if this panel could stimulate more conversations, that would be great for, I guess, from our view.
Yes, Kaspar, go ahead.
I see that Hari and Robert and others are here, and I was very excited by the sort of machine vision based segmentation and classification, and one way that we've seen that you can get over lower SNR is as long as the information is there to be had, using some of these more advanced machine vision models. What do you see the horizon for that, given your work, Lou?
Right, that's a good question. So we are more of the end user of the machine learning algorithm. Actually, I also have a lot of questions on that front. I think you're definitely right. There are actually already papers out there for basically on the spectroscopy level, how the algorithms to basically decoding those Raman peaks in biological samples. And on the imaging front, there are actually papers out there trying to improve the signal noise ratio based on machine learning algorithm and also the imaging speed with machine learning. For us, we mostly use it for imaging segmentation, so that would allow us to sort of get a pseudo fluorescence specificity with a single Raman input. I think that's certainly a very powerful front, would allow us to, with more data analysis, would allow us to get more information. But I'm also being very careful here with the details that we can get from a machine learning, I guess mostly because we're also an end user. I would say for biological applications, especially for those fine features and the very small signal features, we're being very careful for using machine learning to interpret those structures and also features and dynamics, because that's typically where we get some of the interesting biology going on. But how we can improve the quality of the prediction, that's something I think definitely worth looking into.
Oh, can we bring Hari on? He has a question or a comment.
So just kind of following on, one of the things that Kaspar was hinting at, I'm just curious what you guys think about sort of the instrument, right? I mean, so to what extent can you buy more by optimizing the instrument? And, is it the case? So there are things like lenses and objectives and so on. But I'm also curious about your thoughts about kind of parallelization strategies, right? I mean, is there anything that can be done on the speed or sensitivity front that you can think of with better instrumentation? And are people thinking about that? Thanks.
Yeah, that is a great question. So I guess by using a different instrumentation, we and other groups in the field could, I would say the sensitivity now is already shortness limited, which means we need to beat the quantum noise to further improve the sensitivity, if not by doing more averaging to lower this by lowering the speed. Just my on pure the sensitivity front, just my 2 cents, I guess. One of the things to think out, I guess, just to think out aloud, is to see if there's a better laser source, such as the laser using quantum entanglement. That's more of a wider thought that could allow us to bypass the quantum noise. So that's, I guess, on the fundamental limit. On the other fronts, like upgrading of the lenses, that would allow us to have higher throughput and maybe to adjust the resolution. So I think there have been some efforts trying to figure out, how to increase the speed, to do faster imaging, to do hyperspectral imaging, and also to do more quantitative imaging, so that we can do frequency modulation to get rid of, in theory, SRS is quite quantitative. In biological analysis, it's strictly linear to the analyte concentration. Usually what we see as the background is just a rather homogeneous background, but admittedly there are some nonlinear process going on at the same time of the SRS process that would serve as a background. So we can do frequency modulation to get rid of that background. So I guess to make the analysis more robust, there are basically a few ways out there that could be integrable. But again, in terms of sensitivity, I really think it's at the current, maybe there's something I haven't thought about, but on the current setup it's actually quite good toward the quantum limit. So that's something, I guess, if we want to break that ceiling, we need to really think outside the box. But that's a great question.
How big a factor are detectors? Is that something that needs investment?
Detector.
Yeah.
That is, currently it's just currently we just use a large area photodiode. I guess to further increase SNR, you can, for small signals, you can use the, I guess, more sensitive detectors. But there are also limitations because it's usually a trade-off between how much light you can detect on detector versus the so that's one factor, the use of basically the laser power you can use on the sample, and then versus what is the response speed of the detector. So I guess for now, it's very case by case dependent. So it depends on what kind of signals we want to look at to discuss more on the detector part.
Can we bring Luke on? It looks like he has his hand raised.
Yeah, Hari disappeared, bummer. So I wanted to bring it back a little bit to what Alison was asking about this machine learning stuff. So is it the case that you just like download some code from someone after you read a paper, or is there like a real community? And this is a question for Hari and Lou and others and Robert. Is there a real community between, people who are, building scopes and taking images and machine learning folks, or is it more, download my code and try out my algorithm thing? Is there something, we could do to grease the wheels of collaboration between those two different types of science?
I mean, I think it's nascent, Luke. I mean, in sort of biomedical imaging and microscopy. I mean, where it seems to be a lot more developed is in medical imaging, because a lot of these guys have been thinking about these issues, right, for a long time. I'm thinking of the CT and the MRI folks, right? I mean, who have it seems like they have like a decade head start on everybody else. So I do think there's more that can be done there. I mean, right now it does seem a bit like, okay, somebody publishes a paper, you download their stuff, you try it out, sort of see what sticks. Yeah. And I don't know, to kind of bring in our speakers for the session, I don't know what the efforts are like in this space, right, of chemical imaging, if it is just kind of an individual lab basis, or I'm not really sure.
So maybe to wrap up, let's kind of take it back to the biologists. So if a biologist has heard this talk and is really interested in applying this type of microscopy, what do they need to take into account in terms of sample preparation, like the types of questions they can answer? I mean, a lot of people here don't just work in cells or mice, but they're working in Drosophila and in zebrafish. Do you have any perspectives on some of the sort of classic model organisms that we use often here?
Right. Before we run out of time, I actually want to just take back a bit to the probe design before I answer that question, if that's okay. Sure. So yeah, so I guess we have discussed during the discussion session a lot of the instrumentation optimization and also biological questions. But I really want to emphasize is the probe development. I think that's where fluorescence community and also other communities has benefited a lot with the chiming of the booming of chemical biology. But chemical imaging community actually would benefit a lot if we can do more smart designs. Since we have seen, the invention of GFP and then the photo switchable, photo activatable GFP have, made this so much booming to the community. I think for us, we could also benefit a lot from the smart design of either small molecule targeting as an imaging group, and also the larger ones with higher sensitivity for the resonance part. And with that, I think with higher sensitivity and specificity and also multiplexicity, we would definitely expand the biological questions that we could image. So for now, I think we are very good at imaging lipids and also total proteins and also the small metabolites, including glucose and amino acids that I've shown in the talk. But with a better availability of the probes, that would allow us to do higher sensitivity and more specific targeting to, for example, a specific category of the lipids or specific proteins. That would definitely help improve the application a lot.
Do you have any final words you want to add, Lixue?
So yeah, probably I just add a few sentences on the biocompatibility. So for traditional IR techniques, I think the best, the roadblock is the water background. So I have very high hopes on this new emerging techniques with the photothermal and the photoacoustic detection, especially for photoacoustic, because it is known to have in vivo applications. So let's see what's happening in these things, and I'm very enthusiastic on their developments.
Great. Well, so this closes session 1. I want to thank our speakers again for their wonderful presentations. It's really thought-provoking, and and thanks to Kaspar for helping moderate this discussion with me and everybody who participated. I believe what's up next, correct me if I'm wrong, Janine, is a short break.
Correct. Yep. We will resume at 12:25 with session 2.
Thank you.
Sweet.
All right, I think we can get started with session 2. And for that, I am going to introduce Hari Shroff from NIH. So take it away, Hari.
Great. Well, thank you, everybody, for attending our second session today. My name's Hari, and then along with Leong at Janelia Research Campus, we're pleased to chair this session. We have an amazing group of speakers. So Leong, would you like to introduce our first?
Yes, thank you, Hari. Welcome, everybody. Our first speaker is Elizabeth Hillman from Columbia University. Elizabeth, please take it away.
Thank you. Can you all see my slides okay? I know we weren't supposed to say that, but
yeah.
Okay.
Yeah.
Right. So thanks to the organizers. This is it's very exciting to be able to be here. So as a little bit of background for those of you that maybe don't know what my lab works on, we're going to be talking primarily about fluorescence microscopy, so different than what you were just hearing about, the beautiful label-free imaging. So we've developed a technique that does very high-speed, three-dimensional microscopy. It's a form of light sheet that uses a single objective lens. And we've spent the last sort of 5 years, well, 5 to 10 years, really trying to identify all the different ways in which we can apply this to look at all kinds of different aspects of sort of in vivo cellular physiology at the whole organism level, at the organ level, and at the cellular and subcellular level, as well as looking at things like both structure and function. And another sort of aspect of what we've done is that we license this technology to Leica Microsystems, and we've spent a great deal of time over the last 5 years really trying to solve a lot of these problems about how do you actually get technologies out into people's hands, how do you disseminate it, how do you help other people build these techniques in their own lab or host them in your lab. So I'm very happy to sort of share all of our experiences with all of this kind of technology development. Of course, this slide always crashes my computer, so let's see if I can get it to advance here. Okay, so a few sort of more specific things that we've been focusing on a lot lately, sort of in response to where science is going. We have a much more concern over cell identity these days and cell type. And so this is an example of data from a system that we've developed that does very high speed spectrally multiplexed imaging to do sort of 3D imaging of freely moving worms, but resolving many, many different colors. And this was driven by a project with Oliver Hobart looking at these neuropal worms that have very specific colored labeling of exact cells that are then identified within the worm connectome. And so this has been a big area that we've been pushing so that we're not just imaging things quickly, but we actually have cell type, cell identity, and as much multiplexing as we can. We've been trying to do as well a lot of sort of very high throughput imaging of structure, which is really driven by collaborations in expansion microscopy, where you have these very big samples now that are actually very slow to image using conventional microscopy methods. As well, we've realized that our geometry is really ideally suited to looking at things like organoids, things on a chips, things with microfluidics. And again, thinking to push this beyond just say looking at, you know, how something is growing over time, but to think about being able to interact with this living sample, looking at, for example, ways in which it responds to a drug, or ways in which it responds to a physical deformation or flowing things across it, or how cells actually move and change as they're growing in these kinds of versatile in vitro systems. My lab also does something, a whole range of other things, and here's an example of data that we collected in the Taiwan JR gecko mouse, which is doing wide field imaging the entire cortical surface of the mouse in real time, looking at neural activity as the mouse behaves. We're also done a lot of work looking at neurovascular coupling, which is the relationship between neural activity and blood flow, so capturing the dynamics of flowing blood. Now, I put both of these in because we really believe in the lab in looking at things over multiple scales and really thinking about, for example, the environment that a cell might be in, and how crucial it is to sort of consider those different multi-scale aspects of the cellular environment, rather than just thinking of them in isolation by themselves. So that sort of brings me to my slides here, where I've tried to sort of formulate some sort of framework or questions. So most of you usually see this triangle. We think of this in terms of microscopy, right? It's this sort of age-old problem that we can either go fast, we can go high resolution, or we can have a large field of view. But being able to sort of find a happy medium between these, you usually have to sacrifice one for the other. So hopefully I've sort of just shown you by some of my examples that what we've been doing in the lab is trying to bring these closer together, trying to actually have to make fewer compromises so we actually can do, you know, high resolution, large field of view at high speed, so we can start to really look at the intersection of these different things. But as I was thinking about this, I realized that this is also something we can think of in relation to cells themselves. So if we think about a cell has a lot of time, a lot of speed, a lot of time scales itself. So for example, neurons fire on the scale of, say, a single millisecond, right? And so obviously we want to be able to capture things faster and faster. When we first launched Scape and we were imaging at 100 volumes per second, people said, oh, nothing happens in cells at 100 volumes per second. You don't need that. But I'm quite sure that there is stuff that's going on. We just haven't seen it. But also, you know, people live for 100 years, right? And so it's really, really difficult to actually image a cell over this sort of time scale of years, right? And so I think we have to think about this real importance of understanding how cells work, not just on very fast timescales or slow timescales, but that entire continuum in between. The same is true if you think about length scales. And so, you know, this work that I showed you about this wide field imaging, what we're observing here are these kind of ensemble properties of these cells. They're talking to each other over long distances and they're influenced by each other and they're interacting, where if you just zoom in and you're looking at them by themselves in isolation, you're really missing a lot of the information that they're getting. It's like this massive social media going on, and yet you're focusing on one single phone. Bad analogy. We're also really interested, as you can see in my lab, on sort of thinking about cross species. So, you know, if we understand how something works in one species, does it carry over to another species on a different scale, or does it not? And if it doesn't, then why not? Another thing, so then, you know, what we ideally then want to be able to do is really capture things on many of these scales, right? And so, for example, as I showed you, you know, really capturing cellular level, you know, ensemble scale, and then again, all in the context of things like behavior. And real time is so important because, you know, there's been this departure, I think, from thinking about how we want to characterize the way, you know, a cell always does something. You do something over and over again and you see some sort of average response. But the very nature of life is that things are constantly changing, and there's no cell that's in the same state, and there's no system that's in the same state from one moment to the next. And so really getting imaging methods that can capture things in real time that don't require averaging is really essential. And so, you know, we can think about really trying to record lots of things together, like, for example, signaling at the same time as movement, many different cell types all at the same time, I think, if we're going to gather this rich information. But then what's really key here is to recognize that we really need to be ready to analyze it. So I'm going to speed up even more here a little bit because I have a few more slides. So talking about environment and state. So I think I've already touched on this. I mean, this is an example from my field of neurovascular coupling. We can look at blood vessels and relationships in vivo in a living rat brain here, but a lot of the literature in this field actually came from brain slice studies. And in brain slice, you don't have perfusion pressure, you don't have normal levels of PO2 or pH. And so we need to really, really consider this environment, this dynamic environment that's constantly changing. And we can try to recapitulate that in vitro models, but really understanding what we're trading off in these different sort of environments is really essential. And I put homeostasis here. One thing you learn when you're trying to do in vivo work is anything you try to change, it tries to change itself back. And we have to really think about that very carefully because that's actually an important piece of it. I'm mentioning mechanical, you know, mechanobiology is huge at the moment, and we have to think about the fact that cells themselves can exert forces, but they also sense forces. And so that's a big piece that we have to remember. And then I think we have to also really look at the way that cells respond to environmental changes like heat, light, pressure, sound, drugs, and so on. And then, of course, we've got to think carefully about developmental state, aging, diseases, and things like neuromodulators. And I'm going to put a picture of a uterus up here so you remember it, because I think there's a lot of sex disparity in the way that we're doing research these days. And I think there's vast amounts that we need to learn about the way that things like hormones and sex play into our biology as humans and disease. Quantitative analysis. So prospective observation is something that we do a lot in the lab. I think is really key to formulating new hypotheses, but it requires a really diverse skill set. So when we started this project on these crawling larvae, the first thing we did was we took these movies and we spent like 2 years analyzing them, trying to figure out what on earth we were seeing in there, right? And it requires this incredibly diverse skill set where you're going to understand how did the microscope work? What's the biology that you're looking at? And then how do you actually physically do that analysis and go around and around? And I think that that's really essential. I think people have a tendency to sort of search under the lamppost, design one experiment to do one thing, but I think we have the capacity now to do so much more. I think core facilities are helpful to some extent, but I worry when you have this separation between sort of the person that has the idea and the person that knows how to do it. And I also think AI is also a real double-edged sword. I'm sure that's going to come up in the discussion as well. Making things too easy for people, I think, is a huge challenge. So I really just want to make a big shout out here for diverse training, interdisciplinary collaboration. And I think we really need to overcome these disconnects between the people planning the experiments, making the imaging tools, and really sort of asking the biological questions and analyzing the data. And then finally, advanced imaging technologies. I fear that many of the new methods that get developed are just simply too complicated. Applied laziness is one of Ed Boyden's ideas. If it's too hard, don't do it. Wait for something else to come along. I think it's really important that reports of techniques include honest explanations about the level of difficulty to do that technique. If you're climbing Everest, is this the top or is this just the first little foothill and are you going to then have to go even further? I think we need to be clearer. Maybe even editors need to apply some sort of complexity penalty or a warning label to technologies that get published to say, don't try this at home. And I think the user community therefore needs a much better understanding of when something can easily be built, when something probably should be bought, or when something should sort of stay in the lab that developed it, and that lab should be supported to share that technology. So I would just say, just for the record, I think current models and expectations for this sort of tool sharing is pretty broken right now and something that needs to be addressed. And then finally, in terms of large-scale projects, Janelia has really contributed to things like Mouselight, the Genie program. I think that Janelia is really well placed to do some amazing things. There's a big push at the moment to do things like image every single cell in the human brain, for example. We can already image a lot of things in very large and mostly intact samples, and we can now leverage big data methods to make sense of all of that data. But I'm just curious, to pose these questions, what are these projects that we should embark upon, and how does one plan and decide what they are? Who should do them and how? And can we do better than data sharing? There's a tendency at the moment to want to collect a bunch of data, but then just sort of share it. And I think that's another question to consider. And finally, I will just say diversity and equal opportunity is absolutely essential. With that, I just want to credit all the work of my lab and my funding, and thank you very much.
Great. Well, thank you very much for that provocative talk, Elizabeth. That was great and exactly what we hoped for. I guess I'll ask a question since you raised it, this issue of training and skill set. I mean, can you give us some insight more specifically in what you think is lacking there, for somebody that wants to deploy these tools, right, and use them? What is your sense as to what's missing? And
I certainly think, so I'm in biomedical engineering. I originally trained in physics. I think that, in biomedical engineering, what we do is we train people with a very diverse skill set. They have to take biology, they have to understand things clinically, they have to learn the math, the physics, the chemistry, all of those things tie in together. And then in neuroscience, you have people coming in from engineering. I think certainly biology courses should include a lot of programming, a lot of math. I think that we really need to create sort of integrated scientists who are comfortable. I've taught programming to many, many people who came in the room who said, I can't program, and who walked out loving it and thinking it was great. So I think that more sort of interdisciplinary training there is really important. And some of my work on the neurovascular coupling touches on fMRI, functional MRI. And I think one of the most damaging things that happened to fMRI was people trying to make a nice, comfortable toolbox where you just sort of had to load your data and hit a button and it would give you a result, right? It feels great to be helpful to people and make things easier, but when you then separate them away from the actual nitty-gritty of what's happening to their data, It's damaging, and it ended up very much damaging the field, and we got the dead salmon paper and so on. So I'm just desperate for that not to happen in microscopy.
Leong, did you have a question? Yeah.
Yeah, I do. Great talk, Elizabeth. So I'm actually very curious about your idea of core facility being a double-edged sword. So clearly, I actually see your point, and I kind of want to sort of probe a little bit about where do you think the sweet spot is, because you clearly have to balance between technology dissemination, but optimal use of the newly designed microscope, right? So where do you think the sweet spot is so that it is not a double-edged sword?
Yeah, I mean, I think, so, you know, what happens in my lab is, say, a collaborator will come and want to collect some new data on a new sample, and you know, they'll get something good and they'll want to come back. And at some point I have to say to my students, you know, or my trainees, you know, stop doing it for them. Like, step back and let them push the buttons, let them align the sample, let them start to, you know, it's difficult, right? Because you're there and you sort of want to be helpful, and you say, that's okay, I'll analyze the data for you, I'll send you something tonight, right? But that's not helping, right? And so I think it's just that mindset, you know, growth mindset, as my kids' teachers say, right? That you want to make sure that people that are coming who want to adopt these new technologies are coming with a willingness to learn and become independent and autonomous with these technologies, and that people that are sharing them have also that teaching mindset, as opposed to trying to just do it for them. So that's the complexity sort of with the core model. Of course, cores are driven by the fact that a lot of these machines are super duper expensive, so they have to be shared, and then they have to be managed, and they have to be run, and that's a whole other piece of it as well, which is why I sort of favor simpler, less expensive technologies that can become more custom. But it's that mindset, right, of not wanting to be helped but wanting to be taught, that I think we should try to foster.
Okay, thanks. We should probably move on. Leong, you want to introduce our next speaker?
Yeah.
Thank you.
Yes, our next speaker is Robert Prevedel from EMBL. Robert, please take it away.
Yeah, hello everyone, and thanks for the organizers, or thanks to the organizers for inviting me to speak here. So my lab at EMBL focuses, as Elizabeth, very closely on developing new optical imaging technologies, especially for non-invasive imaging that enables to study biological processes in the context of their physiological and natural environment, ideally in an intravital and in vivo fashion. And therefore, the new research area here for the cellular physiology surely sounds really exciting. And so today I want to sort of present my perspective and vision for what type of new tools might be beneficial to advance the understanding of biology in the context of this new research theme, and especially how more recently we now try to visualize the physical aspects of life, and thereby we want to aid biologists in understanding more complex multicellular behavior. So I hope you also see now my laser pointer. And so I wanted to start by basically saying that in this audience, I probably don't need to spend much time on highlighting the fact that optical microscopy is really powerful. I mean, we've just seen beautiful examples by Elizabeth, but also here taken from Janelia, where we can use, for example, light sheet microscopy to closely track the spatial positions of every cell in a living organism with high spatial and temporal resolution. And that of course allows us to visualize everything that we can label with fluorescence. And now increasingly, more recently, this ability is now being combined with so-called omic technologies such as single-cell RNA sequencing, which allows us to capture the interrelationship of gene expression, cell morphology, and the position within live tissues, and also to, for example, create then canonical models of digital embryos, as you've seen just before. And based on such data, then lineage history can be obtained of every single cell throughout development. And that, of course, tells us how gene expression, biochemical signaling is related to the spatial organization of cells and cell types in a developing embryo. But this traditional approach, I think, also fails to consider what many here call the dark matter of modern biology, because in addition to genes and chemical signaling, which we can now easily label with fluorophores of our choice, also physical forces such as membrane tension, cell-to-cell adhesion, but also material properties of the cells and the environment of the cells. Here, especially the elasticity and the viscosity of the cells and tissues do play an important role in tissue function. And so this field of mechanobiology has been flourishing over the past years, as many of you probably know. And it's been fueled by many seminal experiments, such as one that I just want to highlight here by Engler et al., who actually 15 years ago already showed that in the process of stem cell differentiation, the stiffness of the environment of the cell is decisive in sort of driving the cell to differentiate into different cell types, such as neurons, muscles, or bones. And so measuring and visualizing such forces and mechanical properties, I believe, is key in order to gain a full and more comprehensive understanding of how complex multicellular action is orchestrated in 4D and how this sort of eventually gives rise to tissue and organismal function. But the problem that I feel we currently have is that many tools that biologists have face important limitations. And this is because standard methods such as atomic force microscope, micropipette aspiration techniques do not provide really access to the interior of the sample. They only measure at the surface. Other techniques that are based on optics, such as elastography, only have very poor resolution, certainly not on the cellular level, while other methods rely on invasive introduction of particles. So all of these physically perturb the sample in some way, one or the other, and they're overall quite limiting in their performance. So, I guess it would be very nice if we could think of ways to directly image such mechanical properties in the same way we do with a fluorescence microscope, and thereby, benefit from the many advantages that light can offer for us in microscopy. And so luckily, indeed, this is possible with a new emerging technique that's coming out now, which is called brilliant microscopy, that I want to talk about in the next few slides. So this is a really label-free technique that exploits the phenomenon of brilliant scattering, where light interacts with subtle material vibrations called phonons that are present in all matter at room temperature, certainly, as long as they're not at 0 Kelvin. And so then here the scattered light gives rise to a unique spectrum where the shift as well as the line width of this brilliant scattered light depends critically on the sound velocity of the material. And from the sound velocity, we can then deduce material properties such as the storage or loss modulus, which are again connected to properties such as stiffness and viscosity. So our aim was to sort of establish this new methodology in biology. And for this, we set up a so-called confocal brilliant microscope in our lab. And its function is really based on two highly dispersive devices called wipers, which allow us to separate the light in a sufficient manner in order to detect this tiny brilliant contribution from the overwhelming background light. And so our spectrometer now achieves really high optical resolution given by basically the diffraction limit, and also very high spectral accuracy. And in order to really sort of use this system in biology, we built it onto an inverted microscope and added an incubator and a home-built fluorescence confocal. And so with this prototype now, we can clearly map mechanical structures on the subcellular scale. For example, here it's the nuclear envelope of this stem cell, but we can also resolve tissue scale maps of developing organisms. For example, here you can see the zebrafish larvae tail. And especially what we've done here more recently is we characterized and measured a very thin extracellular matrix layer in this fish. And we could actually resolve this as a unique spectral peak here in the brilliant scattered spectrum, and thus measure the unique frequency shift of this tissue layer. And so we hope that this, as well as all the other published work of us and other people in the field, now clearly show that brilliant microscopy can indeed measure mechanical maps with very high spatial resolution, and also in a range of different tissues and model organisms. But one of the big problems is that our confocal brilliant microscope, as well as also other implementations in the field, currently have two important limitations. One of them is the low speed, which means that we are mostly restricted to two-dimensional planes. We cannot image in 3D, while phototoxicity of the laser illumination can lead to cell damage, especially when imaging over long time frames. But both abilities are, of course, really important for any biological studies. And therefore, we more recently set out to develop a new brilliant microscope that exactly address these important shortcomings. with the goal to actually enable live imaging of sensitive embryos during development. And so I want to share some of our preliminary work in this direction. And without going into more technical details, I want to show you on the left the mechanical platform that we've built up for this new brilliant microscope that we specifically built to be compatible with the requirements of live imaging of small organisms, especially embryos. And so it enables mounting and maintenance of small embryos in well-controlled environmental conditions. And on top of our new brilliant spectrometer also features a self-built fluorescence light sheet modality, so that we are able to correlate the mechanics that we measure to any underlying molecular information or cell morphology. And so here on the right, you can see some preliminary first results of this new spectrometer, where we image the mechanics of mouse embryo development. On the left, you can see the light sheet, on the right, the Brillouin. And then what you can see here is mouse embryo development from the morula to the mid-blastocyst stage, so spanning over 2 days of development. And while, of course, the image quality is still, I would say, rather poor and work in progress, I want to highlight that this is really the first live imaging of sensitive embryos with Brillouin. And I also want to highlight that now this gives us a lot more information, namely mechanics in 3D and over time, so in 4D, which we can also correlate to the underlying molecular information. And just to again sort of highlight a little bit the performance, we have a bit more than 1 micron resolution, and the speed is on the order of about 10 to 12 minutes per volume, which is 100 times faster than the previous implementation, certainly enough to study the mechanics in real time. Another beautiful example I want to share today of a traumatic morphogenetic event that is very likely to have a strong mechanical component is in Drosophila embryo development. And probably many of you have seen fascinating movements like this one that were taken by light sheet microscopy, where you can see the process of ventral furrow formation that's going to happen here, where the tissue sort of enfolds and then sort of invaginates. And this is a really rapid phenomenon. But again, by sort of focusing to really this area where the invagination happens, our brilliant microscope is now fast enough to also follow these dynamics in real time and to visualize both the brilliant shift, and which is sort of a proxy for tissue stiffness, but also the cell outlines here in which you can see here labeled with a membrane marker on the order of 2 minutes per volume. And again, here just a few key time points that I wanted to highlight and to show that certainly we do see changes here that correlate with the invagination phase. And we now have to sort of properly interpret these signals through targeted molecular perturbations. But I'm quite optimistic that in the future, this ability will help us visualize mechanics in 4D with high spatial temporal resolution. And so just very quickly, of course, this is an exciting phase of discovery, but there are, of course, fundamental and technical problems that we still need to be addressed. Some are on the order of that Brillouin scattering is well understood from a physics point of view, but not so much in biology. The other one is, of course, technical, where phototoxicity, speed, and sensitivity are still to be improved. And most importantly, we need to find ways to also image other physical properties, such as the refractive index and density. But I also wanted, sort of, looking forward, I wanted to share my view that I feel this new research team of 4DCP could really benefit from a more concerted effort to advance in general biophysical instrumentation and new probes to visualize and characterize the physical landscapes of cells and tissues. I mean, so far, every lab does their own thing. They basically have their own tailored solutions. But I think a unified push would really help the whole community. And here, Janelia could make a huge difference on terms of new instrumentation, along the lines I presented, but also especially on maybe new probes that allow us to visualize forces and tension with new fluorescent probes that they can then be coupled with the typical high-resolution fluorescence modalities that we all know about. And so I'll leave this here with the last slide, because I think I'm going slightly over time. So I think the key challenge that I see for the future is, can we build somehow 4-dimensional physical maps at high spatial temporal resolution to sort of answer key questions in biology? So I hope I didn't go much over time. I want to just highlight and thank my lab and all the funding. So thank you very much.
Thank you, Robert. In the interest of time, we have two questions. Maybe Hari, if we can ask that question, those two questions.
Sure.
So the first question is from Alan K. Can Brilliant microscopy be used to measure membrane tension?
Yeah, so I think I want to be clear that that Brilliant microscopy measures a material property, and so membrane tension is of course a physical force. So directly we cannot measure it. No.
Okay, and then the second question, there's been some discussion of what exactly Brilliant microscopy measures, water content versus elasticity. Do you have any insight into this topic?
Yeah, so I think there was much discussion about this in the literature, and I think there's also been work now that clearly show that it's not just water content, but of course we are measuring the overall material property that's in our measurement volume, right, inside our point spread function. So if this is sort of a highly hydrated, let's say, tissue, then of course this will be dominated by water content. But I think the sensitivity of Brilliant microscopy is certainly good enough to tease apart, even in a not fully hydrated tissue, to tease apart other components. And that has been, I think, shown quite convincingly in the past. Yeah.
Okay.
Thank you. I think with that, the time is up, right, Hari?
Okay, I think we have one more minute, so maybe I'll just ask a few more questions. Have you calibrated Brilliant microscopy with AFM?
No, but there's sort of ongoing efforts in that direction. I mean, again, one of the problems here is that in AFM, we are only able to probe a surface, and we need to use some specific models to maybe extrapolate to larger depths. While, I think the huge advantage of all optical techniques is that we can really probe inside and with a three-dimensional resolution. But of course, the other point is that I had briefly on the slide is that AFM measures the so-called Young modulus, we measure the longitudinal modulus. They are slightly different in their definition. So we don't expect a sort of a one-to-one correlation, but I think it's also important to maybe more systematically establish these sort of correlations and study those. And that again, this could, large-scale efforts could be beneficial in that direction. Yeah.
Great. And with that, I think we're out of time. So Leong, thank you, Robert, for that great talk. And
thank you. And our next speaker is Zhuoran Ma from Stanford. Zhuoran, take it away.
I want to thank you, Leon, for the introduction. Hi, I'm Zhuoran Ma from Professor Hongye Dai's group at Stanford University. So today I'm very excited to be here to talk about our work on in vivo molecular imaging in the second near-infrared window, or the near-IR2 window, which is basically an optical window from 1000 nanometers to 1700 nanometers. So I will first talk about some background and motivation, so why we are interested in doing imaging in this novel wavelength window. And then I want to give an example to show how we can really using near-IR2 imaging to visualize cells in vivo. So fluorescence imaging is a powerful tool in cell biology. And here's a table of some of the most commonly used fluorescent probes. So as you can see, most of these probes emit in the visible window, which is from 400 to 700 nanometers, or the near IR1 window, which is from 700 to 900 nanometers. Unfortunately, fluorescence imaging in those wavelength ranges could not give a very high spatial resolution, especially in deep tissue. So here is an example. So suppose we have some fluorescent molecules emitting at 650 nanometers. And if they were covered by a tissue of 2.5 millimeter depth, you will only get a very low resolution image like that. And that is actually mainly attributed to the scattering of photons in biological tissues. And to understand how we can circumvent this problem, we need to take a look at the scattering behavior of different biological tissues. And the tissue scattering can be quantified by this parameter called the reduced scattering coefficient. And it has been shown that this parameter scales inversely proportional to wavelengths for many different types of tissues. So in other words, if we want to get a better image resolution at a deep tissue penetration depth, we can simply do imaging at a longer wavelength window. And here are some real examples. So here we want to image the brain vasculatures of a mouse. So if we do imaging in this traditional near-infrared window, we can actually get a very blurry one. And if we do imaging in the longer wavelength near IR2 window, you can start to see those vessels more clearly, but still the background is still a little bit high. further, we can do imaging at an even longer wavelength window from 1500 to 1700 nanometers, and we call it the near IR2B window. So now you can really see all the vessels clearly with minimal background. So this result basically tells us that the near IR2 window, especially the near IR2B window, is a better choice for in vivo fluorescence imaging. Now I want to show you a real example. So the biological problem that we are interested in is the interaction between tumor cells and T cells after immune checkpoint blockade therapy. So here is a very simple overview of the biology. So some tumor cells overexpress this protein called programmed death ligand 1 or PDL1. The interaction between PDL1 and PD1, which is expressed on the surface of T cells, can trigger some downstream signaling pathways which deactivate the T cells. And that's the reason why T cells cannot respond to tumor cells in many cases. And people have discovered that by blocking this interaction with the antibody of PD-L1, we can reactivate the T cells so we can attack the tumor cells. And that is actually the basis of immune checkpoint blockade therapy. And it's very important to visualize the interaction between T cells and tumor cells in order to come up with a better theranostic method and also to understand the underlying mechanisms of the tumor immune interactions. And people have been using things like ex vivo immunofluorescence imaging to do so, but those kind of methods could not provide a dynamic information of the distribution and interaction of cells. We can actually use near IR2 imaging to do a better job. And to do so, we first synthesize some near IR2 nanomaterials. So the first one is this lead sulfide, cadmium sulfide core shell quantum dots, which has a very narrow band gap, so it has a longer emission wavelength. And the second one is this erbium-based nanoparticle, which has intrinsic photoluminescence in the near IR2 window because of the electronic transition between different 4F energy levels of the erbium ion. So here are the emission spectra of these nanoparticles. And you can see both of them emit in the near IR2B window. And the interesting thing is that we can actually differentiate their luminescence by lifetime imaging. And here's how it works. So if we excite the materials with a laser of 800 nanometers, only the lead sulfide can be excited. On the other hand, if we excite them with a laser of 980 nanometers, both of them can be excited. But the erbium nanoparticle actually has a much longer fluorescence lifetime compared to the quantum dots. So we can detect the signal after a delay of 1 millisecond to totally get rid of the signal from lead sulfide. And by this method, it really allows us to label and image two different biomarkers at the same time. So we first conjugated the erbium nanoparticle to the antibody of PD-L1 and then injected this bioconjugate to a CT26 bearing mouse. So the CT26 tumor is a mouse model of colon cancer, which overexpresses the PD-L1. So you can see the accumulation of the erbium nanoparticle in the tumor region over time, which can be quantified by this by the signal ratio between the tumor area and its surrounding normal tissue. So you can see the tumor to normal tissue ratio can reach up to 40 in this case. On the other hand, if we just use a free unconjugated nanoparticle, the accumulation inside the tumor region will be much weaker, so which can be seen by this largely reduced tumor to normal tissue ratio. And an interesting fact is that the antibody can retain its therapeutic efficacy after being conjugated to the nanoparticle. So here you can see the tumor was eradicated 20 days after the injection of erbium nanoparticle anti-PD-L1, which basically indicated there might be infiltration of T cells in the tumor area. So now we want to really visualize the T cells there. So what we can do is we can conjugate the lead sulfide quantum dots to the antibody of CD8, and then inject this bioconjugate to the same mouse 2 days after the injection of erbium anti-PD-L1. So again, you can see there will be accumulation of the lead sulfide in the tumor area, which is shown by the green color here over time. And also it gives a very high tumor to normal tissue ratio of about 30. On the other hand, if the mouse was not treated with anti-PD-L1 antibody, the signal in the tumor area will be much weaker. And at the same time, the signal in the liver and spleen will be much stronger. So we can actually use a signal ratio between the tumor and normal tissue, or the tumor and spleen, to characterize the infiltration of the T cells in the tumor microenvironment. If there is activation and infiltration of T cells, both ratios will be much higher. we can also image the tumor microenvironment at a much higher spatial resolution with the help of our home-built light sheet microscope in the near IR2 window. Here is a very simple diagram showing how it works. So basically, we utilized two objectives. So the first one was used to generate a light sheet which selectively illuminate a very thin layer of the sample. And the second objective lens was used to detect the fluorescent signal. So by moving the stage, we can basically scan the whole sample and then reconstruct the 3D images of the sample. So here is a video taken 2 hours after the injection of lead sulfide and anti-CD8. So the red regions here shows the tumor cells which are labeled by the Erbium nanoparticle. And the green color here are the lead sulfide quantum dots which are still circulating in the blood. And you can see that there are some green spots moving around in those areas, like those ones, which basically shows the migration of T cells in the tumor microenvironment. We can also do a 3D reconstruction of the whole area at 24 hours after the injection of lead sulfide and anti-CD8 to really visualize the distribution of T cells and cancer cells in the tumor microenvironment. So again, the red color here shows the PD-L1 positive cancer cells, while the green color here shows the CD8 positive cytotoxic T lymphocytes. And you can see there are some overlapping between those two colors, which are those yellow color here, which shows the interaction between different types of cells. And by scanning a larger area, which includes both the core of the tumor and its peripheral regions, we can see that there are still some T cells in the peripheral region of the tumor, which really shows the infiltration events is coming from somewhere outside the tumor area. All right, so to summarize, hopefully through this very short talk, I've convinced you of the advantages of doing in vivo imaging in the near IR2 window. And in this very simple example, we've demonstrated that we can actually image the cancer cells and T cells with the help of our near IR2B probes in physiological. However, there are still a lot of challenges that remain to be solved in this area. For example, we need to develop materials with better optical properties, such as longer emission wavelengths, higher quantum efficiency, and narrower emission bandwidths. And also, we need to develop some functional materials which can respond to things like signaling molecules or the membrane potential. And also, by integrating some other microscopy techniques or image processing algorithms, including those artificial intelligence-based image processing algorithms, we can really get a better performance for near IR2 imaging. So we have some recent works on those areas as well, but due to the limited time, I didn't cover it here. So you can check those papers if you are interested. But with the advancement of materials development, microscopy techniques, and image processing algorithms, I really believe near IR2 imaging can be a powerful tool for probing cells in physiological conditions. I want to thank my advisor, Professor Hong Dai, and also the lab members and our collaborators. And also I want to thank NIH for supporting our research. All right, thank you.
Great, thank you, Zhuoran, for that nice talk. So maybe I'll start with a question. I mean, I think your application sort of nicely show that if you want to image some structures in an animal, right, with a fluorescent dye, I mean, there may be no other way if you want to go deep inside a mouse or something than using these techniques. But I'm just curious as to your thoughts about sort of somewhat more modest length scales, right? Suppose you just want to image a few hundred microns in. I mean, where do you see your techniques fitting in versus say 2 or 3 photon fluorescence microscopy, and what are the bottlenecks to kind of bringing NIR2 into sort of the more sort of tissue scale rather than whole animal scale imaging?
Yeah, that's a great point. So we also have some work on the imaging those tissues instead of the whole animals using our NIR2 techniques. And in our case, I think one advantage of our method compared to things like 2 photon, 3 photon imaging is that we don't actually need a very complicated optical systems. For example, we only need a very simple laser. For example, we can even use a very low power continuous wavelength laser instead of those femtosecond lasers to excite our samples. Yeah, so for tissue imaging, we can also get a very deep penetration depth. And also I think another point is that we can actually combine our NIR2 imaging with 2 photon microscopy. Yeah, so that would give a even better result in some cases. For example, we can use 2 photon So we can do excitation in the near R2 window as well. So yeah, so that might be better.
Leong, you want to ask a question?
Maybe Robert raised his hands first, so I'll let Robert go first, and then I'll.
Thanks. Yeah, so really nice talk. I was wondering about this cellular specificity labeling. Can you sort of specifically label particular cells? Can you functionalize those probes and maybe use, I don't know, halo tag, snap tag?
Yeah, that's a great point. Yeah,
labeling, is that possible?
Yeah, I think that's possible. Yeah, we can. Yeah, I think our method is pretty compatible with traditional labeling methods. Yeah, but those areas remain to be explored. But yeah, I think that's definitely a very good direction to go.
So Zhuoran, your ability to tune the lifetime of the fluorophore actually is something that I'm actually very interested in.
Yeah.
In addition to having scattering, like Harry is saying, is if you go deep, you have light scattering that you're trying to solve. But even if you don't go deep, there is tissue autofluorescence background left, right, and center in most tissue.
Yeah.
Right? Is it possible to tune your the lifetime of your fluorophore to the point where the excitation light and the detection timing are not synchronous, so that you turn on the light, you excite, and then you turn off the laser, and a split second later you detect, so that all the autofluorescence would have been decayed by now?
Yeah, that's a great point. That's exactly what we did here. Yeah, so we just turn on the light for a very short period of time, and then we just turn it off, and then we can detect the signal after some time. So we can totally get rid of the signal.
That's actually a very powerful way of getting rid of a lot of autofluorescence, especially when you at some point when we're going into a high fluorescent background, like liver, or even looking at blood, for example.
Yeah. So I mean, for those kind of lens night-based probes, so they tend to have a long fluorescence lifetime, which really allows us to do this kind of lifetime gated imaging. Yeah. So.
Okay. And there is one question, so I think we can address one question before we go into the discussion, Harry. Are the lifetime measurements time correlated or in frequency domain?
It's not in frequency domain. Yeah, it's just time correlated.
Okay. All right.
Okay. Great. Thank you.
Yeah. Thank you.
So maybe we should just go into our discussion.
Yeah, I think the timing is perfect.
Yeah. All right. Great. So I guess Leong, do you want to start us off, or?
You have the slide. Why don't you go ahead? I'll let you.
Sure. Yeah. So I guess one question that I guess is also something we discussed a little bit in the last session is to what extent, I mean, for all three speakers, I'm interested in your thoughts about machine learning and to what extent these optical problems have to be solved optically, right? So maybe Zhuoran, why don't we start with you?
Yeah. Okay. So that's a very good point. So actually, in one of our recent work, we demonstrated that by machine learning methods, we can using fluorescent probes emitting at a lower wavelength window, but still get a very similar spatial resolution to ones I just showed you. So this is just by some image-to-image translation algorithms using machine learning. Yeah. So I think and also I do believe that machine learning can help us with analysis of the distribution of those cells. But one question is that we need to be careful when we apply those machine learning algorithms. For example, in many cases, those machine learning algorithms are just, especially deep learning algorithms, are just like a black box. So they don't have a very clear interpretation. And also, it's still a problem whether they can be generalized to other systems, which is not seen during the training process. So I think those questions are still need to be answered if we really want to apply machine learning to get some reliable results. Yeah.
Mm-hmm. Robert, do you want to go next? I know you've done some recent work in this area.
Yeah, I mean, I can only fully agree, right? I think if we need to find ways where we can also, check the black box to make sure that sort of what it outputs is sort of trustable. Yeah. I mean, I don't want to point to a recent paper that we had, but I mean, if we can find ways where at least sort of partially we can acquire ground truth on the fly, this would be really needed, you know. I think we are at a, like, let's say, a dangerous point in time where, you know, if too many people use AI methods without sort of questioning them too much, they just sort of go wild and let them apply to their methods, you know, it's going to be difficult to really then afterwards tease apart to which extent has the black box hallucinated something, to which extent does it really capture true biology. Yeah, that's my opinion.
Yep. Elizabeth, do you have anything to add?
Yeah, I fully agree with everything everyone said, and I think, you know, I said it too, that we have to be really careful and we have to have really good interdisciplinary training. I think one experience I had was I had a lot of data scientists who wanted to collaborate, but they wanted to just grab a data set and run off with it. And I think that we need to educate them as well and really bring them in. That's another thing I said about data sharing, just sort of dumping data on a cloud somewhere and hoping someone's going to come and take it. You know, we've got to be integrated with them. They've got to understand the properties of the imaging system. They've got to understand, you know, the complexities of the experiment. And the most key thing is, like, what are you actually trying to learn? You know, what is the point, right? It would be so foolish of us to ignore this as a technology that could really, really help us. It's like having a car in the garage and walking to the store. But on the other hand, you can't use it for everything, and you have to be very careful and cautious. And maybe, you know, once you know what you want to do with it, you use it to do that thing 1,000 times. So yeah, we need more integration and engagement of that community, and really bring them in and co-train them, or learn it all ourselves, which is hard because it didn't exist when we were learning.
I think that in a way, I think that in a way lead us to the second topic that we were thinking about discussing, is when is imaging alone insufficient? So Elizabeth kind of brought up the point that there's a lot of information extraction that is required. Clearly, we have to think about, you know, how we can deal with this deluge of data and to extract the information. And we were wondering how much of it is in the hand of the biologist and how much of it is in the hand of the instrument developers, or how much of it has to be co-developed together, and how much of a pre-warning, as Elizabeth said, has to be given to the biologist before they even, you know, thinking about touching the microscope. Any takers? Guess not.
Okay.
Well, maybe we can go.
Yeah, maybe the only thing I can add to this is, I think, you know, instrument developers can only do as much, you know, about the instrumentation, let's say the engineering and physical aspects. I think it's always key to really closely involve biologists, and I think Janelia does this really well, EMBL as well, bringing together people from different fields. And also we as tool builders, we have to sort of educate biologists so they understand limitations and potential problems, artifacts, misinterpretations of the tools, you know, that we are building. And I think that can only be done by sort of providing the right environment and having people really closely interact with each other.
So I guess I have a question, you know, for Elizabeth, based on something you said. You brought up this nicely, this kind of pyramid of frustration, right? That optical microscopists think about, and then you started relating that to the physiology. So I guess starting with you, I'm just curious, like, what is the bottleneck as you see it right now? I mean, you know, for example, let's take your instruments as an example. I mean, is there something that is sort of a routine bottleneck, or is there an axis that needs to be further developed, I guess, optically, right, when you study these physiological systems?
Yeah. Well, for light, it's always scattering. It's always depth into the tissue, right? And I've worked at all scales. I did my PhD doing optical tomography with diffuse light, and then now to microscopy. And so, you know, and we keep hoping that, you know, Luke's going to make the near infrared dyes that we need to get deeper. That's the biggest challenge. I mean, we're actually pretty good on signal to noise now. You know, the cameras, the lasers, the dyes. You know, the animal models are beautiful. You know, but light scattering is the thing that's in our way of really getting, I think, the sort of high-speed holistic organ level imaging that I think we would really like. So at the moment, it's a bit of a sort of bootleg, you know, a bit over here and a bit over there approach.
So then maybe, you know, following up on that for Zhuoran, I mean, you know, it seems like there do exist some dyes anyway, right, that are quite a bit better, right, in these different windows. What is the, you know, sort of my naive question to you would be, what is the bottleneck that is just preventing everybody from using these technologies all the time? Is it the dye? Is it there aren't enough good dyes, or does the instrumentation suck? I mean, what is the limiting factor, Zhuoran?
Yeah, I mean, there are questions in both areas. So for example, for the instrumentation, if we want to do near IR imaging, we need some specialized cameras. For example, in our case, we use a indium gallium arsenide camera, which is kind of different from what people have normally used for imaging in the shorter wavelength windows. And for the dye, yeah, so typically the fluorescent, yeah, I think it's still largely unexplored. So for the for those dyes in the visible window or the near IR or the near IR1 window, they usually have a very high brightness, for example, a very high quantum efficiency and narrow emission band. But for the fluorescent materials in the second near infrared window, they just typically to be much dimmer. So they have a much lower fluorescent quantum yield, and also they have a broad emission, so there will be overlapping between different colors. So it's hard to do multiplexing. Yeah, so there are definitely a lot of challenges that remain to be solved in the chemistry point of view. Yeah, so I think that's something that we need to still working on.
Leong, you want to ask a question, or I have like a million questions.
No. We have a million questions on our slides, but we can always bring them up. Should we go to the next topic and then
Sure.
come back and maybe wrap up with a bunch of questions?
Please, yeah.
All right. So the next thing that Harry and I put on the slide that we thought we will bring up is, can the microscope adapt to the biology and not the other way around? So at the AIC, for example, we turn away a lot of really cool projects simply because the biology cannot adapt to the microscope. And I know that there are a lot of development that has been doing precisely that, to try to, you know, your best to adapt microscope to biology. And I wonder if the panel or anybody wants to chime in a little bit of some of the, you know, latest things that are being worked on. We certainly have a few ideas around, and maybe we'll wait to see if anybody want to chime in first.
Looks like Elizabeth had her hand up.
Yeah, I mean, I was just going to say that this is another count against the core facility, right? Because, you know, if you can have a technology there, you can customize. It can be in your own lab, and you can make the adaptations that you want to study specific things. Then you know, you're better off, right? I mean, and that's what my lab has always done. We go after studying weird questions and then build the microscope specifically to answer that question, right? But, you know, it's not usual to have both of those sort of expertise in one place, which brings me back to training. So I'm just sounding like a broken record at this point, but I think it's definitely possible, but it requires technologies that are perhaps lower cost and more accessible, and then to be done on a sort of single lab, custom, bespoke.
Certainly, but I think, you know, that I think again is the sweet spot we're talking about, and core facility comes in various flavors as well, from all turnkey instrument to AIC, for example, that can cater to a wide spectrum of applications. So I think we kind of have to calibrate that a little bit. So some of the things that we Harry and I were thinking about is, a lot of time biology have to be tracked by more than just one microscope, especially to sort of come back to the point that Elizabeth made so eloquently earlier, is that you have to track multi-scale in biology. do you have to always perform everything in one scope? And if you can move the sample from one scope to the other with the right fiducials, with the right kind of smart microscope, can we continue to track biology in multiple scales? Is that something that, for tool developers anywhere, and especially at Janelia, something we should think about? So that we can follow one biological question through multiple length scale and as well as multiple time scale, as Elizabeth has also pointed out. And that is also something that was a recurring theme from the tumor microenvironment workshop, for example. So sometimes things happen really, really fast, and then nothing happened for a long time, and then something happened really fast again. And that's biology.
Yeah, Robert, maybe you can comment on this, because you showed an example, this nice example of this new microscope, right? Where yes, you can get sort of forces and fluorescence at the same time, but even then there's kind of a mismatch in temporal scale. So have you thought about sort of this thing that Leong just brought up?
Yeah, no, I mean, certainly I just want to say multi-scale, imaging is something that we certainly need. Maybe I want to go even broader than, or more general than what I presented with fluorescence versus this brilliant microscopy. But I think having it under one microscope is probably the best solution. I mean, you guys in Janelia have built, I think, one of these pretty super cool instruments, that has all the different modalities combined in one. So I think some method of being able to intravitally, sort of follow the organism, the tissue of interest, and then sort of at particular time points maybe arrest, and then maybe even zoom in at higher resolution. I mean, down to the maybe ultrastructure. I think this is something that I see will be more and more needed in the future. And then of course you need to have techniques that bridge these scales, right? We can go from the light microscopy scale down to maybe super resolution, even further into even cryo-EM. So any sort of correlative methods that can actually span the entire spectrum. This will be the holy grail of the next 10 years or so. And another sort of area that I really like is how we can combine functional recordings with sort of underlying ultrastructure. Right? I think there's plenty of examples now coming from the field of neuroscience where you image dynamic activity of thousands of neurons, and then then you sort of stop the process, and then you try to reconstruct at a really high resolution the cellular position and the connectivity between all these neurons. And these are sort of hero experiments that these days are being done by single labs or institutions, but we have to find a way to kind of make that possible for more and more labs, right? So that we can advance neuroscience, biology in general. How to do that, I don't know, but that's certainly the holy grail for the next few years. Yeah.
I think Alison raised her hand earlier. Alison, do you still have a question?
Not necessarily a question. I kind of wanted to push back against this sort of concept that core facilities are like detrimental to good biology, because I think that it's in a it's a little bit detached from some of the realities of scientific funding. I think it's like not necessarily reasonable to expect everybody to be an expert in everything, and so I think one of the ideas behind Janelia was really to put people in the same place so that labs don't have to be everything to all aspects of their science. And so I think, I think that always having to sort of build the thing to do to answer your one biological question, for labs that are capable of doing that, that's true. But like how, the other thing that sometimes we talk about at Janelia is making science accessible. And so that seems sort of antithetical to that mission.
Thank you, Alison. So I actually saw questions in the list that will actually lead us to the next questions that we have, and this is from Zalai, if I say your name correctly. Are any of these methods able to image and track very small molecules like dioxygen, nitric oxide, hydrogen sulfide in real time, molecules that react with these molecules, and then fluorescence are available? something like that. And this sort of, I think, jived very well with the fourth questions that Hari and I put together, which is, can we infer the unseen? Robert has beautifully talked about biomechanical forces. One of them is one of the unseen, not visualizable factor. And then the other one is thermal measurements, for example. Can we increase the sensitivity and resolution of thermal measurement? And the other thing is, again, coming from all the other previous workshops that are mainly presided by biologists that clearly have bombarded us tool builders with a lot of questions, is there are a lot of biological signals that do not occur in huge bursts and fast bursts, but there's this sustained level that is just right around background, and it affects biology long-term, and it affects biology in a very big way. Is there a way that we can see it also not only from the instrument development perspective, but also from machine learning, and more importantly, a probes that can bring this signal above background? So a combination of all of them so that we can actually track those signals long term. Luke?
I mean, I'll just jump in. I mean, I think this is on us as probe builders, right? I mean, you guys can image whatever, and it's amazing, but we're, and part of the reason for this meeting is Janelia historically has thought about things that are really fast, as Elizabeth said, right? Voltage and calcium transients, et cetera. So the question is, can probe developers make super slow, bad things, right? And how do we do that and make them usable? In a sense, making integrators instead of indicators, so we can, image an hour or a day or a month later and get a signal out, right? And I think there are some nascent ideas. My colleague Eric Schreider made this beautiful calcium integrator that was gated with light. I think that general concept could be extended to a variety of other biological processes.
Yes, Elizabeth, please.
So a few things. So, back in the day, we called it molecular imaging, right? And you're absolutely right. We had a probe that, just shone brightly when it was in the presence of whatever, right? And back then, we didn't worry so much about resolution. And I do want to just say that, the obsession with resolution is really getting in a lot of people's way. And, I wanted to then sort of on the largest question, I teach engineering design, right? And when you do that, you start from like, what is the problem we're trying to solve, right? And then you find a solution that matches that. And so oftentimes people will come to me and say, I want to use escape to measure this thing. I said, no, I want you to use wide field. You need an LED and a camera, and that's what you need. And they're like, but I want to use escape. And I'm like, no, you're going to learn what you asked me if you do this much simpler thing, right? And so, I think we all have to take that approach as tool developers, who want to listen, and biologists have to really, I think, bring it back to sort of what is the actual problem we're trying to solve, and be willing to accept a solution that may not be the fanciest, or the, or maybe it can be solved with a better die, but not a better camera, and so on. So I think that's really key. There's really this mindset of people wanting to use the fanciest thing or to get the highest possible resolution. And in fact, if we really focus on what it is that we need to measure, because it's true, measuring slowly varying things is really hard because a lot of our techniques require some sort of background subtraction. But if the goal is to measure something like that, you can look at things like, dynamic contrast that can be done over and over again and track something that's then non-normalized. So yeah, I think maybe even, and I just, before we end, I don't want to say I'm anti-core. I'm not at all. I just saying, I think that there are just perhaps better ways to do it. Maybe collaboratories where you bring together people to have these conversations at the beginning to decide how to solve these problems together, and then go at solving them, so that everybody's kind of integrating their knowledge and their expertise to get to the solution.
Okay, well, thanks a lot for that last comment. Elizabeth had the last word there. I want to thank all our speakers, and thank you, Leong, for doing this as well. And yeah, I think we're on to a break.
Thanks. Yes, indeed, we have a break, and we will resume at 1:40 with session 3. All right, I think we should go ahead and get started with this session 3. Eileen Furlong from EMBL and David Stern from Janelia are our discussion leaders, and I'm going to turn it over to Eileen.
Great, yeah, thank you very much. Yeah, so after hearing 2 amazing sessions on imaging, I have to say I've never even heard of some of those types of microscopy. That was amazing. But now we're going to have 2 exciting sessions on omics. So this is the first one. And I think we should just get going. We've got 3 really exciting talks. So our first talk is from Doug Fowler from University of Washington. So please, Doug.
Can everyone see my slides correctly?
Yes, perfect.
Great. Okay, thank you so much. Yeah, I'll just echo what Eileen said, which is that this was an amazing morning for me to just watch really incredible things, some things I hadn't even heard of in imaging. Very inspiring, very exciting. So my lab at the University of Washington in the Genome Sciences Department focuses on developing new technologies to study genomics, and in particular the effects of genetic variation, which I'll talk just a little bit about, and also how cell internal states, and in particular like chromatin and nuclear architecture, relate to cell morphology and behavior. And so what I'll talk about is, in the frame that we were asked, a method that we've developed for studying cells, and trying to forge that link between their internal state, either genetic variant they might carry, or other features of their internal state and morphology and behavior. And our approach falls into this sort of ex loco or out of place category, which contrasts with in situ methods in genomics or omics technologies generally. So that's my goal for the next few minutes. We were asked though to talk first about or to mention what would be our ideal method. And from an omics perspective, what I would really love to have is a robust set of tools for doing, omics, for measuring properties of cells in either intact organisms or at least tissues, where we could read out at a single cell resolution and with spatial information, at least one, but ideally, many of these different omics readouts. And I think it's fair to say that we're very far away from having such an ideal method. So what we have today, like I said on my intro slide, are methods that break down into 2 categories. So on the one hand, we have in situ methods where we can, as we just learned, can take tissues or even whole organisms and in some way annotate individual cells, in the most basic, realization with a dye or antibody, but in genomics, perhaps with in situ sequencing or some other method. And here we preserve spatial and morphological information in fine detail, but we're limited to one or just a few measurements. On the other hand, we have ex loco methods, and again, apologies for any real Latin scholars. I'm probably not using the right Latin words, but Google Translate hopefully didn't fail me too badly. But anyway, where we can examine cells either in a tissue context or in culture, figure out what their morphologies are, at least in some gross classification-based way, and then recover the cells that we're interested in physically to do something with. And while this abrogates fine detail spatial information and morphological information, it does give us piles of cells that we can feed into any omics assay that we like. And so I view these 2 approaches as contrasting and complementary sort of states of the art. And so we developed one of these ex loco approaches for covering cells based on morphology. And the idea of this method is, okay, we start with some cells either in culture or in a tissue, and they have a diversity of morphologies or phenotypes, perhaps driven by genetic variants that we've introduced, or perhaps owing to stochastic variation or development or some other perturbation. And like I said, our goal is to identify a subset of cells that we're interested in based on morphology or behavior, and then physically recover those cells for study using some sort of omics approach. And in fact, we prefer to do this not just for one subset of cells, but for multiple subsets of cells defined based on their morphology simultaneously. And so we developed this method, visual cell sorting, that's actually inspired by previous work in this space that accomplishes this goal. And the idea of visual cell sorting, it relies on 4 different methods that come together: an automated microscopy and fluorescence activated cell sorting. I won't introduce because everybody probably understands those, or they're easily Googleable. But the other 2 I'll just briefly introduce. So in visual cell sorting, we use a digital micromirror device, which is just an array of very small mirrors that can be switched on or off. And if you put this in the light path of your microscope, and it's, there are commercially available configurations of wide field microscopes that have these devices, by switching the mirrors on or off, you can produce any pattern of illumination that you like. And just to give you a sense, here's the size of 1 pixel in our microscope of our DMD at 20x magnification relative to the size of a cultured human cell nucleus. So with this device, we can illuminate any cell or subset of cells that we find interesting in a field of view. So then all we need to do is record the illumination to allow us to then sprout those cells that we thought were interesting. And so to do that in the first version of the technology, the published version, we use a photoconvertible fluorescent protein called Dendra2 that starts out life as a green fluorophore. And then if you irradiate it with a very small amount of UV light, it undergoes an irreversible photochemical reaction to become a red fluorophore. So said another way, we can use our micromirror device to just zap cells that we're interested in and change them from green to red. And that enables their recovery. And so bringing all that together gives you the visual cell sorting workflow where, we have some cells that have, varying morphologies or behaviors, and we classify them according to those morphologies or behaviors. And here we're using what I think were referred to earlier as off-the-shelf deep learning based classification methods. And then then the microscope is instructed to photoconvert our fluorophore in the cells that we want to recover. And then this process just iterates in an automated fashion over, and just to give you a sense, in a 24-hour experiment, we can examine on the order of 1 million to 5 million cells, depending on what context the cells are in. Then we can recover those cells using fluorescence activated cell sorting according to their photoconversion. And I presented this here as kind of a binary, but in fact we can photoconvert Dendra2 to different degrees and get 4 different morphologically defined bins, or encode a continuous phenotype in the differing degrees of activation of the photoconvertible fluorescent protein. So that's visual cell sorting. So we're using this technology to study how, the sort of cellular heterogeneity that arises upon stress. So for example, drug treatment, and then also large libraries of thousands of genetic variants that we introduce. And we can read out, a variety of different things, including, the identity of the variant, if we're using a variant library, or, various functional genomics or proteomics readouts as well. And so just to give you a flavor in the last few minutes of a few things that we're doing, one experiment involves a drug that we've already heard about today, paclitaxel, which is a microtubule inhibitor anti-cancer therapeutic. And it's been known for a long time that treatment with this drug produces profound nuclear morphological alterations in some but not all cells. So sometimes you get this normal looking nucleus, and other times you get a lobulated looking nucleus. And people have speculated about why this happens and what it means. So we use visual cell sorting to basically take cells, treat them with paclitaxel, and then take this, now morphologically heterogeneous set of cells, demix them, and then use single cell RNA sequencing to ask what the internal state of these cells with abnormal morphologies were compared to the normal, morphologically normal cells. And what we found, I'm not going to go through the, the data because that's not the brief today, but basically what we found was that the normal appearing cells have high expression of many known Taxol resistance genes. And this sort of recapitulates lots and lots of resistance gene finding experiments, as well as clinical expeditions to find resistance genes, and we sort of learned some new things about how this drug works and is interacts with cells, and have hypotheses about how these normal appearing cells relate to the appearance of long-term drug resistance. Anyway, that's one way in which we're using the technology. Another way is to analyze the effects of genetic variation at scale. And here we're focused on a really fascinating gene called lamin A, which encodes the primary proteinaceous component of the nuclear lamina. And variation in lamin A, germline variation in lamin A, can cause a variety of different, seemingly disparate diseases. So cardiomyopathy, muscular dystrophy, those seem, okay, can imagine how those are related, but also lipodystrophy and a premature aging syndrome called progeria. And one of the interesting things that people have noticed about lamin A variants is that many, but not all pathogenic variants that have this altered lamina morphology, where pathogenic variants have this punctate or aggregated phenotype in patient-derived cells. And so we're basically using visual cell sorting to analyze large libraries of lamin A variants, which when expressed or ectopically drive this morphological abnormality if the variant is pathogenic. And if the variant is not, then you don't see that pathogenic phenotype. So anyway, this is just a culture of many thousands of lamin A variants all mixed up, but each cell expresses only one variant. We can use visual cell sorting to sort those cells out and then deep sequence them to score them for the score each variant for its ability to drive this phenotype. And I don't have like the full data set to show you, but in a pilot library, we see that, synonymous variants have sort of normal morphology scores on this end of the scale, and missense variants, some of them have normal morphology scores, some of them have abnormal morphology scores. And this little pilot library of 100 variants contained 3 pathogenic variants that we know about, and those all had abnormal morphology scores. Okay, so what are we trying to improve? Our imaging and throughput, moving into tissues. Now we work on live or fixed cultured or primary cells, and then some of the data integration and embedding challenges that have been talked about before. A middle distance goal that we're driving at is to, in the context of a developing mouse, isolate cells based on their morphology and then apply single cell sequencing, particularly relative to chromatin and nuclear architecture, to unravel how morphology changes in development relative to chromatin state. Okay, with that, I'll just thank the folks in my lab who did the work, in particular Zhuoran and Hyunjin, and our many funders. Okay, thanks so much. And sorry I went over. I tried.
No problem. Thank you very much. So we have a couple of questions already, which I will go to from the audience. So can the visual cell sorting work for 3D tissues? I guess that's what you were getting at, but maybe you can answer anyway.
Yeah. You can definitely perform the method on tissue slices and thin tissue slices. I'm not, I didn't discuss it in the talk, but we've put a lot of effort into methods for recovering intact nuclei from fixed, and others have too, from fixed adherent cells or tissue slices. And, because you can photoconvert a nucleus, you can recover intact nuclei and do visual cell sorting in those contexts. A large tissue volume is extremely challenging because at least we don't have the imaging chops to get Z resolution. You can get, X and Y resolution in a tissue, but in the Z plane, a lot of things get activated, and that's a problem. If anybody has a smart solution to that problem and wants to chat, we would absolutely love to talk with you. But that's where we're at the moment.
Well, I see Gordon has his hand up, so maybe we can go to, maybe he has a solution.
No solutions, just more problems. But I was going to ask, what's the current scope of, there are photoactivatable, fluorogenic dyes. What is the current scope? I was thinking about, how do you bring this from your Dendra system, which is, very elegant and working, most importantly, to, applying this to maybe non-genetically modified models. Are there dyes out there that are useful, you think, or, just curious.
Yeah, did Luke put you up to answering this question? So, this is great because now I get an extra minute to talk about slides I would have put in my talk. So version 2 of this technology just ditches Dendra 2, which has some nice properties, as you pointed out, but does demand engineering, and instead moves to a system where we functionalized an antibody, both with one of the photoconvertible dyes from Luke's lab, and just another dye that lets us kind of normalize the photoconversion signal. It's a photo activation signal, actually, and that has what has allowed us to go into primary cells and dispense with having to do any cell engineering. And actually, the antibody, because it can be labeled to a very high degree, works way better. I mean, you just get cleaner signal across the board. So we've tried to solve that problem. That's our solution. I don't know if it's exactly the right solution, but the reason why having the dye pinned to the antibody is nice is that, again, we can localize the dye to the nucleus. And that's useful because, it's easier to find nuclear edges than cell edges, especially in tissues. And so, you can target the photoactivation to each cell, with higher fidelity if you focus on the nucleus.
Okay, there's a very quick yes/no answer question, should I say? But yes/no answer. Kimberly Beatley asks, do you need the mirror-based illumination system to do the virtual cell sorting?
No. If you want to use a confocal, yeah, but better if you have it, I guess. Thanks.
Okay, good. I think, though, that we should probably move on now to the next speaker. So our next speaker is Emma Lundberg from Karolinska Institute. Welcome, Emma.
Thank you. I'm gonna share my screen. So it's great to be here, and I'm gonna talk about the work that we've done in the Human Protein Atlas and some of the challenges that we're facing, and some of the, I guess my talk will end with a list of the methods that I wish we had, but that we don't. And our work is focused on imaging the human proteome. So not very super fancy imaging methods, but it's at large scale instead. So this work is closely related to the Human Protein Atlas, where we're mapping where all human proteins are in organs, tissues, cells, organelles, body fluids, etc., using immunoassays, microscopy, and our proteome-wide resource of in-house generated and validated antibodies. And I am particularly interested in the subcellular organization of proteins. And we all know that cellular processes are partitioned in time and space. So knowing where and when a protein is present gives us good clues about its function. Also, we know that relocalization of proteins is an important form of functional regulation, or even particularly dynamic cell regulation. And we also know that mislocalization of proteins may be cause of disease. So I think that this spatial information about protein distribution in time and space is greatly underrepresented in systems level descriptions of cells. And this is what I'm going to talk about now. So to start with the easiest part, the spatial proteome partitioning. So we have for over a decade been localizing human proteomes systematically. We published a map with basically a subcellular map of the human proteome, where we localized the majority of the proteins that are expressed in cultivated cells to 32 different cellular structures and could define organelle proteomes. And one key finding here that is very intriguing is that half of all human proteins localize to more than one compartment. As you can see in this image, this protein is in the nucleus and on the plasma membrane. So this is interesting because these multi-localizing proteins may have a context-specific function or even moonlight in different parts of the cell. And if this is the case, it greatly increases the functionality of the proteome and also the complexity of the cell from a systems perspective. And if we think about moonlighting proteins, there's some databases that describe this, and I think the current literature estimates that about 23–25% of all human proteins are moonlighting. I would personally guess that number is much, much higher, maybe even greater than this multi-localizing number. And if we really want to look at this complexity, this is one image. These are genetically identical cells where we stained one protein, enolase 1. And if you look at these cells, you can see that some cells are dimmer, some are brighter. So there's a difference in expression level of the protein. Also in some cells, the protein is in the nucleus. In other cells, it's in the cytosol. In some cells, it's both, and sometimes it's also on the plasma membrane, as you can see here. And this is a well-characterized moonlighting protein, so we know it has completely different function. In the cytosol, it's a glycolytic enzyme. On the plasma membrane, it's a plasminogen receptor, and in the nucleus, it has some kind of tumor-suppressing DNA binding activity. So just by changing the expression levels and localization of this one protein, these cells displayed a great functional heterogeneity, I would say. And this is only one protein. So when we analyze our huge image database, we can see that 19% of the human proteome shows this type of single cell variability. But of course, we work with antibodies, so we're stuck with static analysis, and that's a bit hard when we want to look at the temporal proteome partitioning. So here, of course, there's, temporal variation can range across scales. We see short, like second or minute variations, and there's also longer transgenerational variations, even in cultivated cells. So I think that in all there's need for better temporally resolved assays or better temporal indicators to generate pseudotime measurements, which is something we would love to do with our antibodies. One thing that we could easily do with our antibodies is to build a pseudotime map of the cell cycle. So we've made a map of RNA and protein expression in a single cell study of the cell cycle. And in this way, we could find, as recently published, 300 new cell cycle proteins, many of which also affect the growth when we do knockout studies. And what is interesting here is that most of these proteins are stable at the RNA level. You can see this flat line. This is the cell cycle pseudotime. These cells divide at 25 hours. Whereas the protein profile is, we see a clear temporal profile at the protein level here. And this indicates that these proteins are regulated at a post-translational level. So this also emphasizes the need to not only study the transcriptome, but also to study the proteome. Interestingly, only 1/3 of the cell-to-cell variability that we observe of the human proteome in proliferating cells can be attributed to the cell cycle. So there's a great heterogeneity that we don't know the reason for yet, if it's deterministically determined or what it is. And I also want to say that there's a lot of translocations that we observe here as well, particularly to the nucleus at certain points in the cell cycle. But then if we look at a particular group of proteins, it's metabolic enzymes. So compared to all of the proteome, metabolic enzymes shows great heterogeneity. So if we look at all human metabolic enzymes, 40% show cell-to-cell heterogeneity, most often not correlated to the cell cycle, or in part by translocations correlated to the cell cycle. And again, here we see that there's much greater population variation on the protein level than RNA levels. Again, it hints at post-translational regulation. So here, I just want to show an example. We can see this is an enzyme in the cholesterol biosynthesis pathway. And we can see in our cell line that we study that the known function of this enzyme is in the cytosol, but we can see that it's also sometimes found in the nucleus, and we also see different expression levels. So in order to really, and we see this again for thousands of enzymes, but to confirm that it's true, we can look at our tissue images that we have in the protein atlas, and here you can see that we see the same heterogeneity within glandular cells of the stomach or within acinar cells of the pancreas, both what we think is temporal heterogeneity and this spatial heterogeneity. So to confirm that this is truly temporal heterogeneity, we've been generating GFP tagged cells together with Manu Leonetti at the Chan Zuckerberg Biohub for about 80 enzymes, and taken a single cell and then let that cell grow to see if a single cell can recapitulate this entire heterogeneity that we see. And here, this is that image. We can see that, yes, one single cell can indeed capture this diversity. And this is the case for, I think, 98% of the enzymes that we tagged and studied so far. But then if we think of our ultimate goal is not only to localize proteins, but to understand their function. And in essence, I would say that proteins often exert their function through interacting with something else, another molecule, another protein, or something else. So one way to try to understand cell function is to describe it as in as protein communities or interconnected networks of dynamic protein interactions. And I think that this paper, not from my lab, but it's a great paper that was published a couple of weeks back from the Geier lab at Harvard. They show protein-wide protein-protein interaction networks from different cell lines, and they show that only half of the protein-protein interactions are shared between cell lines. And they show that the core complexes, essential proteins, are these interactions are identical, whereas the cell-specific interactions are the ones that link the core complexes and rewires the subnetworks to create a cell-type specific interactome. So, and in line with this, we've been collaborating with Trey Heidelberg at UCSD, where we try to integrate protein-protein interaction data, which is known to be a bit noisy, with image data, because basically it's measurements of spatial proximity, but at completely different scales, nanometer versus micrometers. So we have used different machine learning models to embed either image data or protein-protein interaction. The spatial measurements to make pairwise protein distance that we can then calibrate physical distances using known complexes. And in the end, we can make this subcellular hierarchy, that kind of typical Trey Heidelberg style. But what we get to here, and I should say this is really only proof of principle. We did this for 700 proteins in HeLa cells, so it's not a full proteome or anything. But what's we build this kind of gene ontology-like hierarchy. So you have the cell at the top, the nucleus, the cytoplasm, and then it goes down to smaller and smaller parts. So we could reveal 69 cellular systems, of which half are not documented before. They are not in gene ontology, and that's all the purple ones. And we have been validating a lot of these. So we know that indeed they are real components in the cell. So we believe that building these types of hierarchical cell models that bridges data across scales, captures pleiotropic effects, so multilocalizing proteins, but also gives protein interactions a spatial dimension and helps out a bit with false positive interactions that are found there. So with that, I think my time is up. And what we need, what I would love to have is, first of all, better spatially resolved assays to image temporal protein regulation to capture both short-term and long-term very heterogeneity. I would be able to measure protein-protein interactions at scale in single cells over time. I also think we need better computational tools to build multi-scale and multi-dimensional models of cells. And if we really want to understand protein function, for example, the multilocalizing protein, the enzyme in the cholesterol biosynthesis pathway, we don't know the function in the nucleus. So I think we need tools to perturb specific protein localizations or specific protein interactions, and not only being able to CRISPR silence the expression, for example, so that we can really understand the different functions that proteins can have. And with that, I would like to thank my lab and funders, and thank you for listening.
Thank you, Emma. That was very nice. Some claps there. We have one question already in the Q&A, and please, for the other attendees, put your questions in the Q&A. I mean, this question, I think you partially answered already, but I will just read it out anyway, and you could maybe expand. So this is from Vad Perutz from ISS in Mexico, and it relates to a little bit what you said about differences in cell cultures and tissues. So how much do you know that the protein localizations that you see in cell cultures map to what you see in tissues? And I think you showed us some of that for metabolic enzymes, but maybe you could answer yourself.
I can absolutely comment on that. In general, we don't have the resolution that we have in the confocal images and the tissue images, so it's hard to say if a protein is really in the microtubules, for example. But in general, it correlates very well with the difference that there's more nuclear proteins in cultivated cell lines, and there's also more nuclear proteins in cancer cells compared to normal tissue. Probably that's related to the proliferative state of the cells, about 10% more, so it's quite significant. Besides that, we see the same degree of heterogeneity in tissues, so we're pretty sure that it's accurate.
So, I mean, maybe I could turn that question around on a more, I think biologically interesting side, because you showed us that 19% of proteins have this amazing heterogeneity from, the cell membrane, cytoplasm, nucleus for that one protein, and you have 19%, but that's just in one cell condition, one cell line. So if you now had that info, if you looked at all proteins in all tissues or all cell types, I mean, do you think it would be, 100% of proteins, or how high up do you think it would go?
No, so the 19% is actually in several cell lines. And most often when we see heterogeneity, we see it for 80% of the heterogeneity, for 80% of the proteins where we see heterogeneity, we see it in all the cell lines that we test. So a lot of it seem to be conserved, but some seem to be cell type specific. But in the tissue images, there's quite some difference between cells that tend to have more variability and cells that don't, but that might relate to subtypes of cells that we can't really tell apart.
Okay, David?
Yeah, I was curious, since you're probing 25,000 proteins, it must be that some of these do not have really well characterized functions, or maybe you've found through your imaging that the predicted function is inconsistent with the distribution that you see in the cells. So I was just wondering if you could comment a little bit on how much the imaging improves our understanding of function and annotation.
I think the imaging helps quite a lot for functional annotations, although we are not the ones making the functional annotations. That will often be other databases like Uniprot or any scientist out there. But I think it's pretty there's really no information out there, that you have to have more stringent validation of our results in order to approve that location.
I think there's a very interesting question also from Kaspar, who's a Janelia fellow, that basically asked, are there similar libraries of validated antibodies for model organisms, so mouse, for example? How big an effort is it to generate that?
It's a pretty big effort, I would say. And there's not any such collection for mouse. However, mouse and human are pretty similar. And the advantage compared to commercial antibodies, where you often don't know what the antigen was, we have made the antigen sequences public. So you can just blast it to your organism of interest and see if you can use the antibodies. I was just checking the other day, in 81% of our antibodies are in theory applicable in elephants, for example.
Okay, good. We have some other interesting questions, but I'm afraid I think we need to move on because it's quite a tight schedule. But we'll get back to them in the discussion. Thank you very much, Emma. So our next speaker is Benedikt Geier from the MPI for Marine Microbiology. And I think we're going to get back to spatial metabolomics. We heard about this in the very first talk today. I can't hear you. Sorry, Benedikt.
Is this better?
That is much better.
Okay. All right. Thank you. Thank you for the kind introduction. I think there's no meeting where the sentence of "you are muted" exists. And yes, I will go back to talking about a spatial component within the field of omics, which is called spatial metabolomics, and which we use to investigate host microbe interactions at a micrometer scale. And the technique, the tool I want to introduce is mass spectrometry imaging. And to give a little introduction, I think if you're developing tools, mass spectrometry imaging, so if you want to study the cellular 4D physiology, I think studying the interactions between cells and microbial cells, so animal microbe symbiosis, is actually one of the biggest challenges and also one of the fields that demands the development of tools. If you look at the human body, we have microbes almost in every tissue, or in many tissues that we have, from oral cavity to actually lung, skin, digestive system. And if if we now think about the system also in terms of developing tools that we want to study cellular interactions, we have to bridge certain gaps where we go from uni to multi, which means we have one host in a whole microbial community. And this is especially if you talk about omics and biomics. And then also we have to go from macro to micro. So in terms of imaging, we actually have to bridge multiple orders of magnitude if we want to comprehensively resolve a symbiotic system. And then what we also have to do, we have to go from histo to chemo. And what I mean with this, so far, we have mainly been studying the histological association from conventional histology down to ultrastructure, how are partners associated, or also in general tissues associated. And now I think we also have to study the chemical histology. And with that, I mean, like Emma said, the proteins within the cells and the tissues, peptides, but also going down to metabolites. And with metabolites, I just broadly define molecules smaller than 1,500 Dalton, which can be amino acids, membrane proteins, lipids, in general, sugars, and so on. And if we now think about a technique that can visualize all of them in a spatial manner, it would be mass spectrometry imaging. And as Eileen introduced me, I come from the Institute for Marine Microbiology, and what we study as study systems is actually a lot of these cryptic species, and we focus on deep sea mussels actually, because the special part about these deep sea mussels is that they can live in the deep sea, and they can only live there because they have symbiotic bacteria that provide them with nutrients. But what makes this whole system so interesting is that they only have two types of symbionts, which is an extremely reduced system. And if you think about the human gut microbiome, we have hundreds of different species of bacteria. And what I want to say with all these weird animals, also, if you think about tools that you think of for certain organisms, you can also think about the organisms that you maybe want to investigate that might be a cryptic species on one hand, but provides a very conserved, a specialized biological function that will really allow you to investigate fundamental biology. So in our case, we cannot keep hosts or we cannot keep symbionts. If you think about this organism living in the deep sea, we have to do everything in situ. So we also have to do in situ imaging. And although deep sea mussels are really far away from all the medical applications that we have been hearing about, if you develop in situ techniques, these techniques more or less direct transfer to if you get biopsies of patients or samples from be it rare infections, infection diseases, for example. And what our big question now is, because we are interested in these small molecules, the small molecules host and microbes interact through. And therefore, we first need to know who is there. So our question is, how can we link the cellular identity of the microbial partners within a host to the metabolic function? And so first of all, we want to know who is there. And the second part is from the same tissue section in a correlative approach. In this case, we want also to know who is doing what. And for microbes and also many cells, what they are actually doing is expressed to their metabolic phenotype, the metabolites they produce. And here's just an example where from the same tissue section, where you have the host nuclei stained in blue, and then you have two different symbiotic bacteria, bacteria 1 and 2 in yellow and magenta, that sit in the epithelial cells of the host. From the same tissue section, we did mass spectrometry imaging, actually resolve different, in this case, there are 3 different membrane lipids, and we can see that on a micrometer scale within hundreds of micrometers, they already greatly differ. But how does it work? So for fluorescence microscopy, we use fluorescence in situ hybridization, which basically allows us to design oligonucleotide probe that binds specifically to the individual microbes, so the partners present in the tissue. And then we can use confocal microscopy to visualize where the partners sit. And then on the other hand, for mass spectrometry imaging, we use matrix-assisted laser desorption ionization mass spectrometry imaging, also known as MALDI-MSI or MALDI-IMS. And this is a technique which is basically among mass spec imaging techniques. It's a very nice sweet spot because it gives you a really high spatial resolution, but also a really good chemical it covers the field of the amount of metabolites, the range of metabolites that you can detect is a really broad range. And how it works is we have a cryo-preserved sample. We snap freeze our samples to basically maintain the chemical and spatial integrity of the sample, and then we cryosection it to also remain soluble metabolites, to have them to remain them in their location. And from there, we take the tissue sections and we spray with an airbrush-like system. We cover the tissue section with an ionization matrix. The ionization matrix basically gives the name matrix-assisted desorption ionization. And what this matrix consists of are small organic molecules that crystallize on top of the tissue and absorb the metabolites from the tissue section. And the beautiful part is now that they highly absorb UV. And if we shoot with a UV laser on top of the sample and ionize this crystalline layer that includes the different metabolites, these metabolites then ionize and then can fly into the mass spectrometer, where we can detect full mass spectra. And in terms of, because we haven't talked about mass spectrometry, what you basically do is you measure the mass over charge ratio of individual ions. And what we can do to turn this into an imaging approach is that we basically spot-wise raster across a tissue. So it's a surface scanning approach. And once we finish scanning a whole sample, each scanning point will turn into a pixel, and we can display individual metabolites that we detected across all pixels for heat maps. And what this means, and really, this is really the core and the power of mass spectrometry imaging, that it will give you an image for every peak that you have in a full mass spectrum, resulting in hundreds to thousands of different metabolite distributions or molecule distributions, depending on what kind of molecular species you want to visualize. And combining these two now in this correlative approach will give you the organism-specific label that you have through the oligonucleotide probes, the high spatial resolution through the microscopy, but then also you will have the label-free mass spectrometry imaging. So as I said, this is really recording the native chemistry of a tissue sample. And because it's so multiplex, this high spectral resolution really gives you, within a single measurement, a lot of chemical information of your sample. And how did we apply this? So if we now look at the same tissue section as I just showed you, I just visualized one of the two bacteria. And if we zoom in, we can nicely recognize cross sections through these so-called bacteriocytes, so host epithelial cells that are filled with the microbial symbiont. And we know that this symbiont produces two types of specific membrane lipids. And these two membrane lipids, they only differ by a single hydroxy group. And we can still see, if we zoom in, that lipid number 1, which is the aminobacteriopan tetrol, which is the 4 hydroxy groups, has a different distribution within single bacteriocytes than aminobacteriopan triol, which is lacking the hydroxy group. And if we overlay them, we can really see that they are spatially different distributed between individual cells, and resolving spatial distribution between two types of metabolites that, for example, only differ by one oxygen or maybe only by one hydrogen group is really difficult, for example, if you want to design antibodies for. And if we now zoom back into the whole tissue section that we scanned, we can see that this is not only on a cellular level, but there seems to be really differences in this metabolic phenotype of these these intracellular bacteria, where we know they are all genetically the same. There's no strain variation, and still so it appears to be that looking at the metabolite distribution, the spatial metabolomics data, gives us information on what they actually might be doing within the tissue. So this really allows us now to look at the metabolic phenotypes of an animal that we sampled at 3,000 meters depth in the center of the Atlantic. And if I now can give a little perspective, I would say that besides all the technical developments what can really make us, so with all these technical developments available, what can really have us better understand the chemical images? Because one thing that we have in the mass spectrometry imaging data now, and this is a picture it took from the latest study that has the highest spatial resolution, where we can really now resolve cells. We can even see, in theory, the nuclei, and here are two different membrane or phosphatidylethanolamine, phosphatidylcholine lipids that are really abundant, and that really, these are the lipids that really make our cells, the cell membranes. So these are essential for us, and now we can visualize them. The problem is that chemistry on chemical distributions does not always perfectly resemble the histological structures that we are used to. And this is why, in this case, the distribution resembles the cells, but it doesn't have to perfectly match. And I think besides identifying the metabolites from the mass spectrometry imaging data, which is a first challenge, is, I think, using correlative techniques with different fluorescence microscopy approaches, for example, can be really powerful. But everything starts actually with the cryopreservation or the sample preparation, as for every microscopy technique. And in this case, this study here was excellent because it really showed that just snap freezing a sample and cutting it and drying it is not enough. So right now, for mass spec imaging, a frontier is really to catch up with the sample preparation, to catch up to the spatial resolution that is now available. And I think, for example, now in Janelia, there's a really broad range of different cryopreservation, cryosectioning approaches that might boost this the sample preparation itself for the chemical imaging. Then for the higher spatial resolution, of course, there's also a lot of expertise in Janelia when it also comes to different laser setups, where the ion intensity or the signal intensity could be boosted, because if you get more ions from a certain spot, you can go with a smaller spot and therefore have a higher spatial resolution. And finally, I think, and this is really missing, and this is completely missing for mass spectrometry imaging, is that mass spectrometry image, that the temporal scale. Mass spectrometry imaging provides a metabolic snapshot that is beautiful and informative, but nothing spatial. And I think if one now starts to connect the chemical identity, the metabolic phenotypes of, for example, a time series from live cell imaging, where samples are frozen at given states, where you exactly know how certain maybe proteins and cellular structures are distributed, and you relate them back to the different metabolites and membrane lipids within the cell, I think this could be very, very powerful. And with this, I want to thank you for your attention. And I also want to thank my group, the department, Group of Metabolic Interactions, and all the funding agencies.
Great. Thank you, Benedikt. Very nice.
Sorry for going over time.
Yeah, a little bit over time, but I thought you were just getting to the sort of juicy meat there in terms of potentially the intersection with Janelia. So I thought it was good not to stop the discussion. But it was very exciting. Actually, can I ask you one question? Because I was very shocked when you said that you could actually see subcellular. Did I get that right? In the symbionts between the metabolites. And I thought, I don't know how big those cells are, but at least from my colleague, like Theodore Alexander, I mean, I thought that the MALDI-TOF, like the spatial omics, metabolomics at the moment was not at single cell resolution. It was kind of, so yeah.
It has reached single cell resolution. So resolutions with pixel sizes, the highest spatial resolution that I just showed is from a lab in Münster. It's between Bremen and Heidelberg, so not far. And they reached 600 nanometers pixel sizes. And another one that we used for the study of these intracellular symbionts is 3 micrometer. But in that case, we cannot resolve the bacteria, but having the correlative fluorescence microscopy changed everything for us. And this is why this correlation between techniques, especially label-free multiplexed chemical imaging, and then going to specific labels, if you manage to match these, it just, when we look at it, we cannot understand the chemical images by themselves at that resolution. If we now have the fluorescence microscopy, it was eye-opening for us.
Okay, thank you. Any questions from anybody else? So there's one question here from Jean, but we'll probably have to move after this one. But it says, how is the specific M/Z corresponding to specific lipid species determined? What if several lipid species have identical M/Z? and 0.3 for the same lipid species.
Right. So if you have the same identifier for lipid species, you will not be able to tease them apart within the mass spec imaging setup. But these imaging setups by now also offer MS2 availability. So if you have enough material of the lipids, so lipids ionize really well, you can really do, and there's a new Nature, there has been a Nature Methods paper where they do on the fly MS2, where they then tease apart lipid species that have the same MOC value.
Great. Well, thank you very much, Benedikt. I think we probably need to move on. Do you think so, David?
Thank you.
Thanks a million. That was great. You're going to stay here. We need you for the discussion too. So I'm joined now for the discussion, co-chairing or co-discussing by David as well, who's also going to join. Yeah, so I guess one of the things that has come up, and is a key for all of us, is the temporal aspects. So I'm wondering maybe if we can start off with you, Emma. Do you see for your methods? I mean, the classic problem for omics, I think, is that we all do, and you were bringing this up as well, Benedikt, is snapshots. So and depending on your snapshot, then you would just think is high temporal resolution, 1 hour windows in the mouse, like half a day windows, etc. So what do you think we could do here? Have you been thinking about this for the protein-protein interactions?
Well,
I know it's a tough one.
Not for the protein-protein interactions yet, but we're thinking of it in terms of rare translocations. For example, when enzymes go to the nucleus at rare occasions and we don't know when. So we're doing some things a bit similar to what Doug was presenting. We call it deep visual proteomics. It's live cell imaging and then an AI that is monitoring the cells. And then we do laser microdissection of the cells where that event happens, or we hope to do so. We're not quite there yet. And then we can do deep proteomics of those cells. So then we would also analyze shape and kind of very similar cells and then certain events as well. And that is one way of getting to, building these continuous, maybe we don't have to care about it being, we're thinking a lot about not actual like real time. Maybe we can work with any type of pseudo trajectory, even if it's not the real time trajectory, to try to understand processes, because that makes it a lot easier from working with antibodies, at least that perspective.
Doug, do you want to add
what Doug is thinking in line with that?
Yes, exactly. So you mentioned multi-omics in an organism, which I would be all for too. But yeah, let's talk about the temporal problem.
Yeah, I mean, I guess my view on that is that we're sort of faced with the destructive nature of all the modalities that we're using, and so it's a little bit hard. I mean, you can do, I think what Emma said is exactly the right thing, and that's what we're messing with too, which is you can build some reporter or whatever, some visual way to read out the phenomenon you're interested in, and you can track cells over time, and you can have some hope of recovering cells when they do something interesting, or have a life history that includes the interesting things that you care about. And so I think that's like, maybe the first layer of answering the question. You could think about ways to, I guess they're increasing the throughput of different stopping points so that you could look at like many cells when they become interesting would be one thing you could push on. And we thought a little bit about that. But I mean, what we really would want, right, is non-destructive measurement methods. And I think for omics, I guess my view is we're sort of hopelessly far from that. And others could disagree with me. Because we're like library screening people. That's one thing that my lab does. So I was thinking a little bit about in Emma's provocative list of things. Okay, well, we're working on ways to tag proteins endogenously in the genome very easily and with different tags. So maybe we could think of a way to tag proteins in a way that we could modulate their localization, and then do a bunch of experiments where we record the, many cells over time and modulate where proteins are. And then at the end, read out, what protein was tagged and what protein we were looking at in the cell. But that's as close as I can come to some sort of non-destructive method. And I think that's, for proteomics and metabolomics, I don't know, there's single cell sequencing folks who would love, in situ sequencing folks who would love to be able to convert like every metabolite to a sequencing readout. But even that is going to be a snapshot. So I don't know, I guess that's sort of an answer.
I mean, I was wondering actually, I mean, maybe it was also what Kaspar mentioned in the first session, you know, that one of Janelia's strengths is protein engineering. And I don't know, what do you guys think about, you know, and maybe you could comment, but for protein, you know, there's only, okay, this is my ignorance as well, an omics person, but, you know, it seems like, you know, there's only a set number of protein folds if you really break it down. And do you think you could develop, like, I don't know, optogenetic peptides that could block or interfere with tertiary protein structure, or, you know, in some way, I mean, how do you get specificity, but, you know, with protein-protein interactions? I don't know. I'm just trying to think, is there ways that we could very dynamically perturb? But sorry, but Emma hand up first and then Benedikt.
No, yeah, I just wanted to say that we've been thinking in those lines too. And even though it might not work for everything, I'm pretty sure that it will work for some things. or for example, being able to, you know, nucleus is easier. We can block import and export, but the readout is not specific, but maybe we can somehow interfere with those type of signals and such. So I still think that is a way to go that is worth investigating some effort into. Then we've also been thinking of, we haven't come up with any solution yet, but it would be very neat if it was possible to, like with Doug's method, monitor cells, but instead of photo converting them into different color, somehow just cross-link it, everything in that cell. Because then we could cross-link cells at certain states, and then you can start to get to protein-protein interactions in certain states, for example. So please engineer something that makes it possible to do that.
Can I bring this around more to the tissue level as well? So
yeah,
you know, Emma and Doug were talking about cells more or less in dishes, I think. And it's not clear to me, but maybe it is clear to you because you've actually dug into this, how much of that heterogeneity actually in a natural setting, in a tissue, in an organism, actually reflects like spatial patterns that make a lot of sense, or is even spatially organized in some way that you could start making sense of it. So I'm wondering, even like without the temporal component, if deep spatial sampling would actually provide much of what we're looking for as far as explaining the heterogeneity in patterns.
Can I start?
Please.
Yeah, yes. So we've been thinking a lot about that, and to some extent, I would say yes. For example, if it's let's say heterogeneity that relates to proliferation, it's pretty easy because you know where you have your proliferating cells, or if it's a random heterogeneity, we will see random cells, and sometimes we start to see clustering. For example, that cells that express this protein in this state are always close to each other. So I think that we can start to at least dissect it into like groups of different heterogeneity by doing like topological analysis of the expression in tissues. But whether we can get to actual, you know, deterministic function from it, I don't know. But at least we can bend it a bit.
I mean,
and try to maybe see which, because if it's random, if it's only intrinsic signals, we're thinking of it as in cell cultivated cells, that we can model this topological view as a model that takes extrinsic and extrinsic factors into account, and at least determine, is this variability coming intrinsically, or is it in part also determined extrinsically? Some things that come
like a really big help there would be is if you could label many proteins in a single preparation, right? So you could somehow take hundreds of your antibodies and label them all differently, and then look at them all in a tissue sample, and then start looking at correlations between expression patterns that look heterogeneous, and start building your spatial proteomics that way. I think that would make I think things with lots that, it's just based on my experience of looking at embryos, the location of the protein in the cell makes a lot of sense once you know the function of the protein. Like a transcription factor gets nuclear localized when it needs to be nuclear localized to drive a particular, and it's cytoplasmically localized when it's not needed in the nucleus. And so there's these things make a lot of sense once they're figured out. But I'm wondering, like I think if you could look at lots of these proteins simultaneously, the distribution of these proteins in a cell, particularly for cells, and I've also seen these patterns as well, where proteins have really heterogeneous expression patterns in tissues for no super obvious reason, just from the anatomy. And so there's something going on internally in that tissue that we're not seeing. But if you could probe for hundreds of proteins simultaneously, you might be able to start teasing out what those
yeah,
factors are.
and we're working with highly multiplexed methods to do that. But I just want to say that there, I think the need is to develop highly multiplexed methods that can also detect low abundant proteins, because most of them that are available today only go for very high abundant proteins, and that's not what we want to look at.
Set of questions, which was
but maybe, sorry, we could.
Oh. Yeah, sorry.
I was just going to pull in Benedikt there, though, because he had his hand up for a while, and I know he was probably also thinking about this.
Thank you, Irene. I think it's difficult to say that we want highly multiplexed, temporal, highly sensitive, but I think what is really key, or what could be really key, is that one uses these spatial omics techniques as really an interface to the highly sensitive bulk analysis. Then you go to the spatial omics, the spatial metabolomics or spatial transcriptomics that link these bulk analysis, but also that provide you with a structure or a really structural component, certain location in a cell, certain co-localization with the organelle, nucleus, whatnot, that then might link to the microscopy, where we have this whole world of spatial imaging over time in 3D. And I think these spatial omics that are currently emerging, it doesn't matter which one it is, they are not as sensitive. They don't have such a high spatial resolution. They don't have any, but they come in between these techniques that we all want. Mm-hmm. And I think if there's a great force to, in a clever way, interconnect these things, that would then give us maybe, or allow us to calculate models or make predictive models on the things that we want, where things are in the cell, and then how they link to a certain phenotype on a protein, metabolite, or transcript level.
Yeah, I agree. But not all of them. You said they were low resolution, but seq-fish, in fairness, is even subcellular, quantitative. But maybe we could ask, Doug there, because Doug, on your sort of wish list, you had your ideal model would be, multicellular organism, you had the picture of the mouse, and multi-omics in there. And I'm just wondering also from your own stuff, I know that you have a genetic variant in there, but how far do you think we're going to be able to integrate multi-omic, like dissociated multi-omics with spatial omics? Like even just, I know this is what people are currently trying, but I guess you must be.
Yeah, well, the brief for this was to be provocative, right? So yeah, the first slide is like the dream that I don't know how to build that machine quite yet, but I guess I would agree with Benedikt that's where we're at now, right? Is we can make these very detailed and fine-grained measurements on cells, and we can do some stuff spatially, and there's lots of smart ways of co-embedding these data sets to make inferences in the tissue context or the intact context. I think that if we sort of think about the history of single cell generally, what we'll learn is something, but I think what will be missed by that type of approach is all the kind of detailed and surprising ways in which things happen in an intact context that don't happen in, the isolated context. And that's where I think, I really like the suggestion, and perhaps Emma's approaches are the most amenable to this, but, thinking about taking advantage either of, massive robotics and lots of money, or some of the the cool like multispectral imaging things that we heard about, but to try to add more labels to intact imaging, maybe there is some hope of really looking at protein localization in a more intact sample. But I guess to summarize, my answer is that I think co-embedding or integrating data from deep single cell, isolated cell approaches and spatial approaches will give us something, but I think we'll still miss the details and then the surprising things that we don't, we're not necessarily looking for. And so, yeah, we're trying it too. I think that's like the next frontier, but it will have its.
Yeah, exactly. Actually, I was thinking also for Leok's Shu's talk earlier, where she was looking at using IR to look at metabolic probes, so basically imaging metabolism. I mean, that I thought would be really cool to combine, Benedikt, with your type, with the MALDI type of approach as well, because you can go more global, but then, image in situ the number of different metabolites.
I think maybe also in combination, because I really love the talk, is that if you have the mass spec imaging, for example, you can check to which extent these types of molecular probes that you introduce to a living system actually alter its metabolism. Maybe it doesn't, maybe it does. It's just sometimes, I mean, have we looked at how membrane lipids, phosphatidylcholine, these lipids not many people are interested in, because they're always there. They're super similar. They're scary to look at because they're exchangeable. They can have same M/C values, different unsaturations. But even on a superficial level, how do these basic things change in cells that we perturb? But also maybe even where we introduce certain chemistry to look at them even in the beginning. I think that goes really down to the basic part, but mass spectrometry might offer a certain tool in that direction.
Yep, I agree. One of the questions, actually, I don't know if can I go to a few questions, David, and because there was one question actually that's very relevant here from a postdoc at CMB, Akski, sorry if I'm pronouncing your name wrong, but it was for a non-destructive approach for protein readouts. Can you track tRNAs, individual ribosomes, and then by changes in the amount of tRNAs around ribosomes would give an idea of the proteins being made? I mean, I guess it's like a bit in situ riboseq. I don't know.
I think that's a kind of a specific style of a more general question of whether there's a generic means of identifying all the protein localization without using tags, without using antibodies. Are there ways of finding the location? Are there features of those proteins? So I mean, another style of this is to use EM, right, and try to model fit the structures that you're seeing, which is starting to make progress with very large proteins. But yeah, I think this is an interesting direction to think about, whether kind of in situ protein sequencing, basically to be able to localize proteins without tags, still be fixed specimens.
Mm-hmm. What about aptamers or approaches like that?
Right. Yeah. No, I think that's in the same family.
Yeah.
Yeah.
I mean, I guess the only issue I see with I mean, the problem with proteins is they're all intermingled and you can't amplify them, right? I mean, all the in situ methods for nucleic acids basically rely on the fact that you can, in a very lossy and crappy way, at least you can amplify some of them. And then once you have those amplicons, you can, sequence them. I don't know. I'm all for protein sequencing is super cool. And I've followed the work from like Ed Marcotte and others with interest, but yeah, in situ, that's a heavy lift.
But I guess once you can convert it, I mean, into, I don't know, into like from protein into DNA, and there's this primer exchange, like immunostaber based on a primer exchange reaction, and then you can amplify in that way. Now it hasn't been multiplexed to a large amount, but in terms of different proteins, but that's something where, I mean, it works with RNA, it works with proteins, and it works because of the amplification possible. I mean, I'm not an expert, but it seems like it can work in tissues and embryos, which of course for me is very important.
And Emma was touching on this idea as well of I think it was Emma, of fixing cross-linking and then detecting cross-link proteins, and those and of course there are labeling methods now as well, but they're very noisy in the data I've looked at, and I think, but I do think there's a lot of runway actually for those methods, for improving, for kind of the engineering improvements in sensitivity and specificity of those methods? And maybe you guys could comment on that. It's not my area, but I just read peripherally.
It's not my area either, but I follow it closely. So of course, most protein-protein interaction studies to date has been done in bulk. So of course, there's a lot to improve, and I think just being able to, for example, cross-link proteins in one cell that is in a specific state, for example, and then sort them would enable us to kind of dissect the PPI data. But I also think right now the main promise of protein-protein interaction data, of course it's noisy, but there's lots of noisy data. Single-cell sequencing data is noisy, but it's completely underused. It's so orthogonal to lots, for example, single-cell seq data. So I would say that it's still, there's really great and big data sets out there. So I would just love to see more computational integration with protein-protein interaction data right now. But otherwise, being able to study, some kind of targeted protein-protein interaction studies in single cells without knowing which interactors you're. Of course, if you know which pair you're looking at, it's easy. You can design many assays for that. But the more kind of unbiased approach or exploratory approaches would be cool.
Right. I think that the fundamental issues there, right, are the temporal and the spatial specificity, right? If you could control the cross-linking reaction to a very short period of time in a very small space, you would dramatically improve, presumably, the signal to noise. But I think that's the area of the limitations right now. But those seem like engineering problems rather than fundamental physical barriers, right? So
yeah, it's up to Luke to sort it out. Yeah, but actually studies have shown that even in cells, but especially in organisms, that when you're fixing like something small like Drosophila embryos, that the cells, of course, on the outside are fixed first, and then slower and slower as the cells in the interior, and yeah, so it's very mixed.
I'm so glad you brought up the fixation issue, because I think most people are not aware of the fact that fixation is disturbing almost all interactions, and there's some really beautiful data on this, on how transcription factors come off the DNA during the fixation process. And so awareness is one thing, but like solving that problem so that things stay where they're supposed to be, or where they were when you started in the experiment, would be really nice. I mean, I think the only solution now is really rapid freezing, which is not practical for anything of kind of interesting size from my perspective. Tissues, right? It'll just freeze too slowly. So maybe you guys have thoughts on fixation as well. I don't know if you've seen any interesting artifacts.
No, maybe we should work not with, for example, direct interactions, but rather measures of proximity in cells, and then knowledge of that the transcription factors falls off, but maybe they're still in closer proximity than other proteins, for example. But yeah, it's a hard problem to solve, I think.
We need high resolution for that. But I'm not sure with the time now. I'm just a bit conscious that there's another session coming up.
Yeah, I think this would be a good place for us to wrap up this particular session. But thank you, Eileen and David, and all the speakers. It's been a great session. I think we will right now just take a very quick 5-minute break. So we will break for 5 minutes and resume for session 4.
Great. Thank you, Doug, Emma, and Benedikt so much. And David for co-discussions.
All right, everyone, I think we will go ahead and get started with the last session today. And our discussion leaders for the session are Zhuoran Meier, Sarah Ruhanafard from Northeastern, and Raj Chari from Frederick National Laboratory. And right now I'm going to turn it over to Sarah as our session chair.
Hey everyone, welcome to our omics session 2. Our first presenter is going to be Shraga Schwartz from the Weizmann Institute. So Shraga, take it away when you're ready.
All right, can you all see this?
Looks good.
All right, great. So I'm really, really grateful for this opportunity to participate in these wonderful discussions. I'm hugely enjoying this format of this conference with the emphasis on big picture and vision and discussion. So in my 10 minutes, I will try to provide a bit of a sense of my field, the epitranscriptome, and talk a bit about our work that our lab has been doing in this area. And then perhaps highlight the key challenge that I believe we are currently facing as a field, and which is severely limiting our ability to advance in it. This challenge has to do with our limitations at the moment in obtaining spatially and temporally resolved measurements. My screen is, I'm having issues going forward. Okay, so as I said, our lab focuses on the RNA epitranscriptome, or on the level of post-transcriptional modifications on RNA. And what catalyzed our interest into this field is a simple, but I think powerful analogy to modifications occurring on DNA and on proteins, where we know that modifications on these molecules can severely alter the properties of these molecules. So for over the past decade or so, we've been asking questions about how modifications in RNA can contribute towards shaping the fate of mRNA molecules. Now we know already, and this is something we've known from years of studies by the entire field, that RNA modifications can have a dramatic impact on RNA. So at their essence, they're a chemical modification, and by virtue of these chemical changes, they can do many things. They can modulate the structure of the RNA. They can give rise to recruiting or repelling of reader proteins. They can modulate the genetic code, which is leveraged by tRNAs. They can impact RNA stability. They can impact translation. And these days, they're perhaps both best known for their roles in modulating immunogenicity. So this is me getting a corona jab, a vaccine against corona, and as many of you are likely know, this is based on modified mRNAs, where all of the uridines within the vaccine were replaced by a methyl pseudouridine, which in this case is used in order to reduce the immunogenicity, the innate immune response, which is triggered by the exogenous administration of this vaccine, and it allows improved translation of this vaccine. Modifications were originally identified in the late 1950s, early 1960s, and they were primarily studied within the context of ribosomes and tRNAs. These molecules are heavily modified. Roughly 5% of the bases on the ribosome are modified. About 10 to 15% of the bases on tRNAs are modified, and there's been extensive biochemical characterization of these modifications and attempts to understand their function. And of course, what made this possible is exactly this, the fact that they are so abundant within the cells that they are biochemically tractable. And as I was getting into this field about a decade ago, I was wondering, what about modifications in mRNA? To what extent could they play a role in modulating the fate of mRNAs? And the difficulty here is that mRNA, in contrast to ribosomes and tRNAs, are not biochemically tractable. So we needed a different approach in order to look at this. And luckily, this was just around the time when high sequencing based methods were becoming available, which we thought we could utilize for this. Now, one challenge we had to overcome is that modifications in RNA become invisible once the RNA is converted into cDNA. With few exceptions, that is typically the case. So we had to come up with approaches which would allow the RNA modification to become visible even after this conversion. And I'm showing here different flavors of approaches that we've developed over time, applied also to different modifications that we've looked at. So on the left here, we're seeing M6A, which I'll talk a bit more about later. This is the most abundant modification in RNA, for which we use an antibody that selectively enriches for fragments of RNAs that harbor this modification. In the context of pseudouridine, we made use of a bulky residue which specifically attaches to the modified base and which leads to premature truncations when this RNA is reverse transcribed. In the more recent work working on N4-acetylcytidine, this is work done together with Jordan, who's part of this session now, Zhuoran Ma from the NIH. What we use is this approach where we chemically derivatize this base. Basically, we reduce it, and this gives rise to C2T misincorporations, again, upon reverse transcription. So all of these methods, they leave marks on the cDNA upon reverse transcription. In all cases, we then couple them to to a library preparation protocol, which then allowed us to obtain transcriptome-wide readouts for these modifications. Now, genomic approaches are very powerful. First of all, as I was indicating, they allow us to obtain transcriptome-wide readouts of modifications. Often they can do so with single nucleotide resolution. In many of the cases, they can even provide us with quantitative or semi-quantitative readouts. So we can estimate the fraction of the modification, the stoichiometry of the modification, the fraction of RNAs that are actually modified. They can also easily allow us to look at the extent to which the modification might be regulated across different conditions. And they can also allow us to study the sequence and the structural determinants that govern the biogenesis, the specificity of the modification. And they can also allow us to look at evolution. So in fact, I go as far as to say that genomics would be a perfect methodology in a universe in which in which cells were basically a bag of RNA and the RNA was inert. That is, there's a fixed amount of RNA and the levels of modifications within it are also fixed and nothing is changing over time. But of course, cells are not a bag of RNA, and RNA is of course not inert. We know that the fate of an RNA is highly complex, going from transcription to processing within the nucleus, and then it can either undergo decay in the nucleus or it can undergo export, and then it gets exported to the cytoplasm where it can get localized and translated, and it can undergo decay. And there are several things that make this very complicated. So first of all, these processes are all coupled to, or many of them are coupled to each other. And especially if you think about this from the perspective of the RNA modification, there's two complicating factors here. One is that the RNA modifications are installed during this life cycle. And the second is that the RNA modifications are impacting this life cycle. And especially when we're trying to understand the functions of the RNA modifications, this can make our life very difficult. So to try to exemplify this, I will provide a sense about this from the context of N6 methyladenosines, M6A, which is, again, the most abundant modification on mRNA. So about 1 in 200 adenosines within our cells is actually not an adenosine, it's an M6A, a methylated adenosine. And it's installed by this complex of 7 different components, the writer complex. There are at least two proteins thought that can actually erase this modification once it's installed. There's an ever-expanding set of proteins that are involved in reading this modification. The machine, all of this machinery is highly conserved and is also pan-essential. So in many systems, when you try to knock it out, you cannot actually knock it out. So we have, and of course the big question is, okay, so what's the function of M6A? And as I was entering this field 10 years ago, basically there was not a single function ascribed to this modification. 10 years later, we're now at the point where this modification is essentially implicated in almost any function along the life cycle of an mRNA, ranging from its production and the modulation of chromatin, going through pre-mRNA splicing and termination and R loops and polyladenylation, in defining its nuclear stability, its ability to undergo export, to localize within cells, to undergo translation, and its stability at the end. And of course, this marks a major advance. All of these studies are incredibly valuable, but there's also a frustrating aspect to it, which is that to some extent, when a modification, the distance between not doing anything or doing everything is actually not that big. So Kurt Vonnegut said this very nicely, that everything in a way is nothing, but with a twist. And what I mean by that is that what I mean by that is that if you're a new researcher these days coming into this field and you're seeing this interesting phenotype for this modification complex, it's really you don't really know where to look at, again, because it appears that this modification is implicated in almost every part of the RNA life cycle. So in a way, we're lacking the logic of this modification. We're sensing a bunch of trees, but we're not really seeing the forest. Now, it's of course possible that M6A does all of this, but at least from a perspective of an Occam's razor point of view, it seems unlikely that M6A is doing all of this. And what appears to me to be far more likely is that M6A may be participating in a subset of these steps, but that the complex and highly coupled nature of RNA biology, within which this modification is deeply intertwined, can give rise to this large set of indirect outcomes. And unfortunately, the limitations of genomic-based measurements makes it really difficult to dissect these questions. And as a consequence, I feel that at the moment, we still lack a fundamental understanding of what this, of why and the how of this modification, why it's there, why we need this massive and essential complex to install this mark. And we also lack some insight into fundamental aspects of this modification. For example, how dynamic it actually is at the single molecule level. And to be able to untangle this, I really think that we need better technology. So if just to finish with the dream technology that, again, I could only wish that we have, that we might have, essentially it would have all of the attributes that we've been talking about also in the previous session. So ideally it would be applicable to live cells. It would give us a readout of M6A at the resolutions at which it actually happens within the cell. So single cell, single molecule, spatially resolved. And when I say spatially, I mean within the with subcellular resolution, temporally resolved, and of course, ideally also transcriptome wide. If we're at to dream, at least we can dream widely. I'll just finish that in 2016's epitranscriptome analysis was deemed method of the year by Nature Methods. More recently, spatially resolved transcriptomics were deemed Nature of the year, the method of the year. And what I really hope is that together these two can combine, can be married to each other to give rise to some spatial temporal epitranscriptomics as a vision for the field. And I'll end with this. Thank you very much, and I'm happy to take any questions.
Thank you so much for a really great talk. I'll kick off with some questions. I have a ton of questions for you, but I'll start off with one. Why do you think M6A of all the modifications has been the one that we've learned the most about first? And just to piggyback off of that question, what would it take to expand to all of the other modifications? There's close to 150 different types.
Yeah. So I'll say M6A. So also when we chose to look at M6A, it wasn't a random choice. I mean, people like to say there are 150 modifications, but in fact, from the early 1970s, we knew that there was one form of methylation that was particularly abundant on mRNA, and that's M6A. So I'd say M6A is by far the most widespread modification on mRNA. And I think that's both why it's the most interesting in a way, because it's so widespread. It's also one of the reasons why it's been so much easier to look at it, even from a technical perspective, because if you're looking for a needle in a haystack, you'll do better the more needles you have and the lower, and the lower the size of the haystack. So in this context, there are many sites to be found. It's simply easier to look at. Other modifications, and we've certainly looked at these, they're much more rare, making it more challenging, and also perhaps more of a question of whether the fact that they're even there is biologically meaningful, or whether it may reflect some off-target activity of enzymes that actually evolved in order to direct the modifications to other targets, such as tRNAs or ribosomes. And I think these are very interesting questions in the field that people are looking at from various perspectives.
Okay, we have a question from the audience from Pangiotis. Do RNAs that stem from pseudogenes get M6A? And if so, does it occur at the same rate as in the regular mRNA?
That's an interesting question. I've actually never explicitly looked at pseudogenes per se. I would imagine that they would. We see them basically in every class of genes. We see them on non-coding RNAs and linked RNAs. So I've never looked at pseudogenes as a class, but yeah, that would be my guess.
Okay, our next question is from Tim Harris. Is knockout of M6A methylase, assuming there is one, fatal to cells or embryos?
Yes, I was saying that M6A is pan-essential. Certainly you cannot make a mouse embryo with a full knockout of M6A. But having said that, you can, embryonic stem cells, for example, they can survive in the absence of M6A. There's nice work from the Hanna lab showing that while they can survive and proliferate, they lose their ability to differentiate. More recently, there's now also, about a week or two ago, there's actually now this drug, the first drug in the market targeting the methylation complex. So that basically it doesn't eliminate METTL3 is the methyltransferase. It eliminates its catalytic abilities. And the idea is to use this drug for leukemia, because it turns out that you can relatively specifically target leukemic cells via this targeting, but without having too much of an impact, one hopes, at hematopoietic stem cells. So there's certainly interest in using it, but if you look, for example, at the DepMap database and you just ask how essential it is, you'll find that it kills, if you try to eliminate it using CRISPRs, it will kill the overwhelming majority of all cell lines that people have looked at today.
Okay, so one more question I'll ask is that you were kind of critical of genomics-based methods to look at RNA modifications, and gave a great analogy about the bag of RNA as the cell. So if it's not going to be a genomics-based method, what would you prefer? Is it a chemical-based method, or what do you think is the way forward? What do you think we need a big development in?
So first, I by no means mean to bash genomics. I love genomics. And if genomics can be in any way combined with additional methods that could also be providing these kinds of resolutions that we're interested in, that would be amazing. I'm a bit like Ben Fowler was saying before, I have this dream, this image, this desire to talk to people and kind of foster this idea and see what kind of things we can think about. And they can be imaging based. Potentially, I mean, especially things like single cell resolution, you can certainly also get at these kind of resolutions also with genomics, even for RNA modifications, and people are already working on these angles. Working out in subcellular resolutions, especially and looking at this in live cells, getting the temporal resolution there will be more challenging with genomics, of course, because of their disruptive nature. So I'm here to stimulate discussions and hopefully come up with ideas.
Okay, and then one last question popped up in the chat. There are usually multiple M6A modifications on one mRNA. Do you think all the sites are important and contribute equally to its control of the mRNAs, or are there differential function?
Yeah, so this is something we've actually, I've shifted a bit in my view of this over the past year or so. I now actually think, so most M6A sites are actually sub-stoichiometric. So even when we talk about M6A on a single, or even when we talk in M6A on a gene, it doesn't really reflect the biology a bit, where we actually have to think about transcripts and whether or not there's a binary M6A at a particular site in the transcript. What we're now actually seeing in our recent work is that when we now have this new method where we're basically integrating over all of the methylation across the entire gene. And what we're actually seeing is that when we do that, and we have this new metric, we can actually we actually get much better agreement between this and different functional data that we've acquired, for example, looking into RNA stability. So we think this integrative levels of M6A across an entire gene somehow best correlates with functional outcomes of M6A. So in that sense, it would almost mean that it's not about an individual M6A here or there. It's somehow about the integral of M6A across the entire transcript determining its fate. That's how we think about it at the moment. We may be wrong.
All right, thank you. I think that we're over time. We're going to go to the next speaker. So our next speaker is going to be Aaron Streets from UC Berkeley.
All right. Can you hear me? Fantastic. So thanks for the introduction, and thank you for the invitation to speak at this workshop. My lab is focused on building hardware and software tools for single cell analysis, and specifically inventing new ways to make precision measurements on multiple aspects of the identity of single cells, and hopefully at a high throughput. But this is something that I want to talk about as a challenge at the end. So how do we quantify the identity of a single cell, or of any cell for that matter? One of the best ways to do that is by looking at the cell with the microscope. We saw some beautiful talks about that in the previous session. And the microscope is indeed the first single cell technique, first single cell tool. And so when we use a microscope, what are we looking for? Well, we're looking for the location and the composition of proteins and metabolites. And we saw some really great ways of identifying proteins with MSTOC and others, and using label-free methods to look at metabolites. And we're looking at their composition and their localization in the cell, as well as stuff like cell morphology. But if we want to get a better understanding of what gives rise to that particular identity, it's useful to quantify some of the genomic layers of cellular identity. So, for example, we might do single cell RNA sequencing. That gives us a comprehensive genome-wide quantification of gene expression, right? Or in some situations, variations in copy number or single nucleotide polymorphisms might sort of inform a cellular identity. But even more importantly, the way that DNA is structured and methylated and organized in the nucleus is sort of, the epigenomic state of a cell is really the input to this state machine of the cell. And one challenge with microscopy is for measuring these other layers is bandwidth. In other words, we can only measure sort of a handful of distinct molecules with traditional microscopy. And of course, there's a lot of distinct molecules in a cell, right? So we have, billions of base pairs, hundreds of thousands of proteins and proteoforms, tens of thousands of genes, and then the dizzying combinatorics of the interactions between all of these proteins and locations in both RNA and DNA. And so for measurements, of these sort of genomic layers of cellular identity, high throughput DNA sequencing has become a really powerful tool for identifying and quantifying molecular composition of cells at the genome scale. Of course, with sequencing, you typically lose spatial information about the cell. And so ideally what we'd want to do is layer microscopy and sequencing-based measurements in the same single cell so that we can infer relationships between genomic information and spatial information. And of course, there's a lot of different ways of doing that. My lab is focused on doing this using a combination of microscopy, microfluidics, and genomics. So we build microfluidic platforms to help us combine microscopy measurements with genomic measurements in single cells. This is actually a stimulated Raman scattering image of a HeLa cell on the left here. And when I say genomics, I'm referring to basically turning molecules into DNA so that we can use a high throughput DNA sequencer to read out their identity. So specifically, I want to talk about a platform in my lab that we've been using to probe the epigenomic landscape of single cells through mapping protein-DNA interactions. So if you take a closer look into the way that the genome is organized in the cell, there's, we know that the genome is temporally regulated, but it's also spatially regulated. And there's, sort of these geographical landmarks that are associated with regulation of the genome. So for example, you have chromatin compartments and nuclear pore complex, and nucleolus modulates ribosomal activation and transcription. You have hotspots of RNA polymerase clustering, and you have the nuclear lamina, which is also associated with repression. And so when you want to identify or map out these geographic, spatial, regulatory regions of the genome, a valuable way of doing that is by mapping protein-DNA interactions, right? So finding the associated protein that you're interested in and seeing where it is. But there's sort of two questions you can ask when it comes to profiling protein-DNA interactions. You can ask where in the genome are these interactions happening, like linearly, what's the genomic loci? And then you can ask where spatially in the nucleus are these interactions happening with respect to other landmarks or other proteins. And so we set out to answer both of these questions in the same single cell using a microfluidic platform. And the tool that we're using to map protein-DNA interactions is a technique called DNA adenine methyltransferase identification, or DAM-ID, a very powerful tool that was developed by Heinekoff and Bosman-Stienzel over 20 years ago. And specifically, we're using it to look at interactions between DNA and the nuclear lamina. So we're mapping lamina-associated domains, which has been one of the very powerful applications of DAM-ID. The way that DAM-ID works is that you fuse your protein of interest, in this case, lamin B1 here, to DAM, DNA adenine methyltransferase. And anywhere that the DNA comes in contact with your protein of interest, DAM will methylate the GATC in any nearby sequence, methylate the adenine in GATC. And so you can specifically digest the genome, you extract DNA, digest the genome with DPN1 that targets the methyladenine and GATC sequences, do targeted adapter ligation, and use PCR to amplify these fragments that represent the protein binding sites. And because we're using PCR for whole genome amplification, we have the sensitivity to go down to single cell. And so Jaap Kind, who now has his own lab, has developed single cell DAM-ID fairly recently. And so using this technique, many labs, including the Van Stienzel lab, have been able to map these lamina-associated domains, which are a really important domain of our genome. There's thousands of them. They represent a large percentage of our genome. They tend to be AT-rich and gene-poor, and genes that are in lamina-associated domains tend to be repressed. And so in addition to using DAM-ID to sequence these domains, you can also image these domains using this trick that Jaap Kind also developed, where he's targeting, using this M-tracer molecule to target a GFP to the genome that has been interacting with the lamina, using this it's a DPN1 restriction enzyme that's lost its nuclease activity and has been replaced with the GFP. So now you can recruit this fluorescent molecule to not lamin, but to areas of the genome that have been methylated. And so in this way, you can not only see like lamina-associated domains, but you can see domains that have been associated with the lamina and have since moved away. And so we wanted to combine these two techniques in a microfluidic platform. So we use integrated microfluidics. These are multi-layer devices that allow us to use pumps and valves to manipulate single cells and to move around reagents and perform reactions with pretty high resolution. So here I'm showing we can trap and sort cells. And so we developed a device called what we call microfluidic DAM-ID. And this is a project that was led by a really fantastic graduate student in my lab who just graduated, Nicholas Altimos, who's now a Hannah Gray Fellow and continuing his postdoc at Berkeley. And so the idea is what I'm showing you here is one pipeline of our device, which is a series of channels and valves where we can trap cells, image them using, in this case, confocal microscopy, but we can also use stimulated Raman scattering or whatever microscopy technique you want. And then sort cells into these chambers where we can perform lysis, digestion, adapter ligation, and PCR, and we can pull out the fragments that have been associated with the genome and sequence them. And so in this way, there's many of these pipelines in parallel on a single device. And so the output of this experiment are single images of lamina maps of single cells. So this is, this is the lamina associated domains, and we chose LADs because we knew where they were. So this is sort of a proof of principle demonstration. We wanted to see if we could see where these domains were, and we knew where to expect them. And then we also get these genomic maps of lamina-associated domains. And so here what I'm plotting is this is bulk DAM-ID. Yellow means that this locus, we're looking at chromosome 1. Yellow means that this locus is associated with the lamina, and blue means that it's not associated with the lamina. And you can see that our single-cell lamina maps recapitulate the bulk measurements pretty well. And we can use a classifier to call lamina association in the 500 in bins of genomic loci here across single cells. Okay, so just quickly, what can we do with this data? Well, one of the things that's interesting is because we have single cell resolution, we can identify regions of the genome that are constitutively associated with the lamina across all cells. And these are also, when we talk about constitutive lamina associated domains, we're talking about domains that are also at the lamina across many different cell types. And we can identify regions that are constitutively not at the lamina across all of our cells. And then we can identify with single cell resolution these domains that are variable from cell to cell. So this represents some variability. And it turns out that these variable lamina associated domains are variable between cell types. And so we compared our data to data from Jaap Kin's single cell DAM-ID paper and showed that domains that were variable in HEK cells had more gene expression in HEK cells than they did in these KBM7 cells. And we can also begin to correlate the genomic lamina maps with the images and cluster cells by their lamina-associated domains and see how their sort of morphological arrangement corresponds to these domains. And so now we're applying this technique to look at really high-resolution laminar perturbations and to see how this is coordinated with lamina association on the genomic loci level. So really quick, just to wrap up, some challenges. So one is throughput. This is not particularly high throughput. We're kind of trapping one cell at a time and imaging it and then sequencing it. And we want to kind of get to this sort of cell atlas level throughput. This is a snapshot from the Tabula Sapiens website. And so we want to be able to generate these data at the same rate that we can do single cell RNA sequencing. And then interpretation, as we start to get more larger data sets with higher dimensions of different layers of genomic identity and images, how do we figure out what to do with this data and learn something about biology? And so we've begun to address these challenges, but there's still a lot of work to be done. We have one technique for barcoding chambers inside the microfluidic device so that we can multiplex. This has been published already. And then we've also started to develop some variational autoencoders. This is for a different type of measurement. This is for paired transcriptome and proteome measurements, but we're starting to figure out how we can reduce this high dimensional space into a lower dimensional latent space, and so that we can visualize cell identity in a much lower space and infer some biological mechanism. So that was published recently in Nature Methods. So we'll stop there. This is we have first two students graduated with their PhD from our lab recently. So we had a small private event. It was very exciting to have after a year and a half of isolation. And I'll take any questions. Thank you.
Thank you so much for a great talk. We have our first question from the audience from David Pellman. How do you discern bona fide cell-to-cell variation in lamina association from noise/PCR dropout, et cetera?
Yeah, this is the most important question. Single cell genomics generally is super noisy. And so what we did was we first made a measurement of background noise by making cells that were just expressing DAM by themselves. And we looked at how likely it is for certain loci to be methylated sort of as background. And then we sort of chose some a set of lamina-associated domains that we knew to be real lamina-associated domains because they've been annotated in other cell types and were annotated in our bulk measurements. And we used this sort of gold set of constitutive LADs and constitutive inter-LADs as a way to generate a classifier so that we could call, how likely is this data actually coming from real association versus background. And we're still working on this. I think we can get better if we can improve like adapter ligation, because that leads to a lot of dropouts, as you say, and sort of improving some of our genomic techniques. But this is a question that always plagues a lot of our analysis. And so we're spending a lot of time thinking about that.
All right, great. I have another question for you. So you mentioned that throughput was kind of your biggest challenge. So can you talk a little bit more about how you might overcome the throughput? What kind of solution is going to be like your first choice?
Yeah, absolutely. I mean, there's a lot of different ways to address this. It depends on how much information you want to get from the sequencing. I'll just go back to this challenges slide. Doug Fowler had a great talk about how you can potentially improve throughput using sort of targeted selection and then FACS, right? Just in the last session. The way that we're doing it is by making a large device and barcoding each chamber. When the limitation in throughput for us is really on the imaging side more than anything, because if you want to image every cell at high resolution, you have to kind of move to each cell and image it. And even if you have a fairly fast imaging modality, I think if you go above 100 cells per chip, it starts to get to the point where you're having cells sitting around for maybe even over an hour on a chip or so. And so I think you can move to higher throughput if you want to decrease the resolution with which you're imaging, but it's a trade-off. I think that there is a niche for lower throughput, higher information content measurements, depending on what your biological question is.
Okay, do we have time for, I think I'm a little confused about the timer. Do we have time for one last question?
Yep, go ahead.
Okay, so we have one last question from Shu. I hope I said the name right. Can you do this with nuclei prep from a tissue sample?
Yeah, so that's a great question, because we end up only evaluating the nucleus. And so we do this for single cell RNA-seq a lot because we're looking at adipocytes. And so we can do single nuclei and we can trap single nuclei in the device, and we can in theory perform DAM-ID on them. Well, although we haven't tried doing DAM-ID on extracted nuclei, but I can't think of any reason why you wouldn't be able to unless lamina association was disrupted. One caveat, however, is that DAM-ID requires a genetic fusion between DAM and your protein of interest. And so we're working on developing some new techniques that will avoid that other challenge. So you could potentially do this kind of approach in primary tissue samples.
All right, awesome. Thank you so much, Aaron. Our next speaker is going to be Winston Timp from Johns Hopkins.
Hi, and thanks to the organizers. I hope everybody can hear me. Thanks to the organizers for inviting me to speak. It's my pleasure to talk to you today about both some of the efforts that we have and some of my ideas about like what would be a good direction to go in to try and probe these questions. And I'm really happy that I get to go after Aaron, who just introduced like some of these ideas I'm going to talk about. So it's really perfect timing. So first, and I know everybody knows this, but I want to sort of introduce the central question that we want to be able to characterize, or at least that I focus a lot on characterizing, is the central dogma. And everybody knows that this is DNA goes to RNA, goes to protein, right? But how this actually happens is far from clear. We have lots of genes in DNA, but not all the genes in DNA are transcribed constantly. And even when they're transcribed, they're not always transcribed at the same levels. A given gene might produce one RNA for a certain period of time, might produce 100, et cetera. And a certain RNA then produces different amounts of protein. and this is things that sort of we've been discussing throughout the workshop thus far, and why it's important to measure this with different angles of attack, different ways of looking at and characterizing what's going on in DNA, what's going on in RNA, what's going on in protein. And so I'm going to argue that one of the ways in which you could try and characterize this is with single molecule type genomics. And this is what my lab thinks about a lot. So we're a sequencing technology development lab. So we've been using newer technologies in long read sequencing that includes things like PacBio sequencing, bio-nanogenomics, and Oxford nanopore sequencing, where you can look at really long molecules. When I mean really long, I mean things that are easily 50, 100 kb, even as long as a megabase of DNA sequencing. And I'll start by talking about DNA a little bit. So why is it good to look at single molecules? I'm going to argue that a single molecule, if you're not doing any PCR amplification, which we usually don't, a single molecule represents a single allele from a single cell. Now, it's not the same as doing single cell because you can only connect information along that molecule, but it's coming at the same problem from a different angle. Here's an example looking at a region where we have three different enhancer regions. Picture on the left is sort of schematic of you have a gene promoter, and then you have these three different enhancers, and there's a couple different conformations that the DNA can actually take. And if we have sequencing reads that are long enough to include, all three of these enhancers, as we do here, and on the top is plotted existing ENCODE data showing that you have certain histone post-translational marks and DNA hypersensitivity locations. You can see here, I have individual sequencing reads. Each row here, this is an integrated genome viewer plot, where each row represents an individual sequencing read that, as you can see here from the scale, is in excess of 28 kilobases long. And the blue locations represent places where I don't have DNA methylation, where it's unmethylated, a CG location where it's not methylated. The red represents locations where it is in fact methylated. And what you can see here is that there's interesting patterns to the methylation, I'm going to argue, where this first enhancer here is largely unmethylated pretty consistently, but the second enhancer is not so consistent. We have sort of heterogeneity at that location. And that heterogeneity, in some ways, you can look downstream and you can see that, like, for example, this read here, I hope you can see my cursor, seems to be relatively unmethylated throughout. Whereas some of these other reads where there's an unmethylated location are coordinated down here with enhancer 3, some of them are not. And one of the things that we're working on doing, and we just preprinted some efforts along these lines, are trying to cluster individual reads according to their epigenetic status. So we can actually look at individual clusters of reads. Now, again, this doesn't mean that outside the realm of the individual read that I can then coordinate that with some other read on some other chromosome. But it does mean that using an almost bulk sequencing approach, I may be able to deconvolute or deconvolve out information about what the heterogeneity of the epigenetic state was, and therefore what the heterogeneity of gene regulation might be as represented by the epigenome. But it's not just that. As Aaron sort of also helpfully introduced, you can also label things like using DAM-ID as pictured here on the right. You can label proximity to another protein, in this case, a nuclear lamina, fused to a DAM methyltransferase, which then decorates DNA that's proximal to it at a GATC location. You can also do things where you're profiling chromatin states. You could just gently lyse your cells and throw in a sort of methyltransferase like CPPI, which will then decorate any accessible GPC sites with methylation, leaving the inaccessible regions blocked by protein or nucleosome, as shown here on the green, alone. We can then, so basically what we can do is we can actually take the DNA and use it sort of as a memory tape. My original training is back in electrical engineering, so I sometimes think about things like computers, where you're actually encoding into the DNA molecule itself what the state of the cell was, what the state of the DNA is, what proteins the DNA was near, if the DNA was accessible or not. And then you can later read that out. And so we've done some work trying to read this out with nanopore sequencing, where we can look at the native DNA methylation, the CpG methylation, as plotted here on the left, and the sort of chromatin state. And this is looking at the X chromosome. The X chromosome in female cell lines is typically very epigenetically different between alleles. And that's why we see these two very distinct clusters, where we have a cluster where it's very accessible, shown here in blue, at the promoter region of PIM2, and then also at this enhancer region downstream, and unmethylated. And then you have another cluster where it's inaccessible and methylated, and we can parse out the difference. We could do allele-specific epigenetic profiling by looking at these individual sequencing molecules. And this makes sense, and it's coordinated with also allele-specific expression that we observe. So we can also try and do this with DAM-ID, and we've started trying to work on this to characterize using nanopore sequencing the amethylation state along the same things, and we see coordination with existing lamina-associated domains. We haven't gotten down to the point where we're able to do clustering yet with this, but we're hopeful that we're going to be able to. But we don't want to just look at DNA. We also want to look at RNA. And so RNA, it's also important to look at single molecules of RNA. Why? Well, because you want to look at the entire molecule to understand the splicing state. If you just use a short read, a small little piece of the RNA sequence, and either using traditional Illumina sequencing or short read sequencing, you're only going to get a little bite at it. And in this example here, if you look at exons 2 and 3, they're basically identical, but exon 1 alpha versus exon 1 beta from these two different splicing isoforms leads to a completely different protein sequence because you have a different open reading frame. You have a frame shift in the resulting protein, and the sequence can be different. And it's hard to read that out unless you're able to, deconvolute or figure out that I have this exon 1, and so this means something different for what protein I'm going to translate. You can also look at allele specificity here. I have, in the case of IFIH1, I have a SNP downstream here, over here in the red box, and then I have an exon that's missing for the maternal allele in the green box, but is present on the paternal allele in the red box, as you can see here in the green box. And I could only tell that if I could have this SNP that's way downstream. And so I was able to sequence the entire molecule. So sequencing the full molecule is important there, but we also want to be able to encode into the RNA molecules information about their state. RNA is sort of famous for having secondary structure. We want to bake that into our sequence that we're reading out. And working with Will Stephenson, we tried to use shape reagents, which actually bind on or modifies the sugar of ribose in single-stranded RNA regions. So you can then later back out and figure out, well, what was the RNA structure of this? This was in a loop area versus a hairpin area of the RNA. And you can actually back that back out. And we're able to see pretty good correlation from doing a nanopore measurement to a traditional shape reagent. And we can do this on a single molecule level. So we can see how this exon over here might influence the structure. And others are working on this too, and have published some really nice work on this, I should mention. And then finally, I'll say that I think we want to also be able to characterize protein, and we want to get to the point where we can do single molecule protein sequencing. I don't have an answer here, but this is part of the question I think we'd like to be able to go after. There's work like ASAP-seq where you use antibodies with barcodes that's been discussed a little earlier. There's work by Marcotte et al. Down at UT Austin where they're trying to do sort of single molecule protein sequencing via Edmond degradation, so-called fluorosequencing. And then there's some work by Jeff Navalla at University of Washington, where he's tried to use nanopores again to try and do protein sequencing. If we can get to the point where we can do single molecule protein sequencing, we can deconvolute how post-translational modifications are on the same molecule. We can get at a lot of questions that are right now very hard to read out. So to summarize, I think to really understand the central dogma of biology, we need quantitative ways to measure the entire molecule, single molecules from each step. This will play a complementary role to some of the stuff that we've been talking about all day with imaging and with sort of single cell methodologies. It's not the same kind of data, but I think together these sort of orthogonal approaches to looking at these questions will yield new insights. Probing the whole molecule without amplification can provide clues to long-range interactions, and we can encode information about the state of the cell or the molecule into the molecule itself directly and then read it out. And it's enticing to think about doing the same thing for protein. With that, I'm out of time and I'll take questions.
Thank you so much for an awesome talk. Could you comment on what are some of the major challenges for nanopore sequencing? How could more people get into this? Just kind of general thoughts on that.
So nanopore sequencing itself, the error rate is sort of always a challenge, but I would argue for a lot of the applications I just discussed, it's not as big a deal. It typically also requires a relatively large amount, especially for the RNA, requires a large amount of RNA to do RNA sequencing. I'm hopeful that will come down. There's been some recent announcements from Oxford Nanopore, the company, that have indicated that they're trying to reduce the input requirements substantially by, say, 3 orders of magnitude. That would be a big deal. And then the computational part is non-trivial. You know, I put some of this data up, but interpreting some of this methylation and some of these modification data for nanoshape, for example, is currently non-trivial. This is something I and many other computational biologists are working to solve. But I would also invite the community to try and look at this. I think that maybe got at some of the problems.
Yeah, so you showed an image of the long reads where you showed 3 different enhancer regions, and you were talking about heterogeneity between the enhancer regions. When you think about the error rate that's associated with your long reads, how do you take that into account, and what kind of controls would you use in an experiment like this?
So that's a great question. Thanks. Generally, when I'm looking at DNA methylation, I don't necessarily rely on any single CG, and the accuracy of methylation calling is up in the area of 95%. This isn't that much better or worse than considering the PCR bias that you might introduce with bisulfite sequencing or other methods. But I would probably try and use as a control some sort of Illumina sequencing to make sure that my methylation frequency lines up, et cetera, in regions where I could. But the error rate, as long as I can map, and as long as I have enough sort of SNPs, if I want to, or mutations, if I want to call genomic variation and phase that, or and it matches broadly what the methylation percentage I expect is, then I sort of buy it at this point. You know, methylation calling using nanopore is now become pretty robust in terms of its accuracy as compared to other gold standards like bisulfite.
Okay, and then you also talked about, you know, every single process that occurs in the central dogma. Do you have a favorite one that you think is like the future of your technology?
I mean, I think that trying to understand, getting a better understanding of how RNA is working. I'm most focused on answering questions in RNA at the moment and understanding how splicing and splicing dynamics is working and how RNA lifetime and how RNA correlates to how much protein you actually make. I think that's sort of a really intriguing question, and I think it's something that would be really beneficial to understand.
All right, well, thank you so much. If there are no other questions, I guess we will shift over to our discussion.
We have the trainee talk. Shuo is next, I believe, isn't it?
Oh, I'm sorry. I'm sorry, I jumped ahead. Okay, so our next speaker is Shuo Han from Stanford. I apologize for that.
Can you guys see my slides?
Yes.
Okay, great. Really thrilled to be here and be the last speaker of the day to wrap up our discussion today. My name is Shuo. I'm currently a postdoc fellow in Phil Beachy's lab at Stanford, working on tissue regeneration and stem cell biology. And today I'll be focusing my talk on some of my previous work on spatial proteomics that I did in my PhD with Alison team. So as we've seen a lot of the previous talks today, we all appreciate that biological systems are incredibly heterogeneous. And this heterogeneity comes both at the tissue level, where different cell types intermingle with each other and communicate, but also at the subcellular level, where different biomolecules are spatially organized and distributed and enclosed in membrane compartmentalized organelles. So while there are tools available, such as single-cell RNA-seq and spatial transcriptomics, that are widely used for dissecting this tissue level of cell type heterogeneity, tools for systematically investigating the polarized distribution of molecules and signaling processes in space and time are rather limited. So to invent new methods that systematically look at this particular level of heterogeneity, TingLab in recent years has pioneered a new approach termed proximity labeling that is able to perform proteomic mapping of a genetically defined subcellular region of interest. So in this method, a promiscuous labeling enzyme, such as APEX, is genetically targeted to a specific cellular region or within a protein complex of interest. Then with the application of a small biotin-containing small molecule substrate and its co-oxidant hydrogen peroxide, the APEX enzyme is able to rapidly generate a highly reactive phenoxyl radical that is able to covalently label endogenous proteins that are only nanometers away from the active site of the enzyme. So following this spatial recording, covalent recording of spatial information in intact living cells, the cells are then lysed and the biotinylated proteins are captured with streptavidin magnetic beads. And the apex neighboring proteome are then identified through LCMS. Using this general concept and workflow, over the years, we have expanded the scope of apex labeling technology, showing that it can not only label subcellular regions, but also molecular complexes, such as the mitochondrial nucleoid, a ribonucleic protein complex that contains the mitochondrial genome. And in collaboration with Howard Chain's lab at Stanford, we showed that the phenoxyl radical chemistry also works with labeling RNA inside living cells. So the approach can also be utilized for generating subcellular spatial maps for RNAs. And then by combining APEX with CRISPR-Cas13 technology, we can localize APEX to the vicinity of a specific RNA of interest in living cells and then capture its interacting protein partners. While these proteomic, transcriptomic, and interactomic studies really sort of demonstrate the utility and versatility of APEX labeling, most of these studies were done in vitro cultured cell lines. So we were motivated by some of the same thoughts behind Janelia's 4D cellular physiology initiative, and we wanted to push forward the proximity labeling technology into contexts or biological systems where things are more physiologically relevant. So in collaboration with Lixue Luo's lab at Stanford, we developed a proteomic workflow that is able to map the cell surface of genetically defined subpopulation of cells within intact Drosophila brain. And for the rest of my talk, I will discuss in a bit more detail about this particular work. So we wanted to develop this method to study the Drosophila olfactory system. In the Drosophila olfactory system, different types of olfactory projection neurons makes precise one-to-one matching with its corresponding olfactory receptor neurons, as shown by this diagram here, in the antenna lobe region of the fly brain, to help establish a complete, precise olfactory neural circuit. And during this process, the cell surface of projection neurons are critically important in mediating neuronal wiring and is ideal for the application of in situ proximity labeling technology because the cell surface molecules are not only dictated by the projection neuron themselves, but also its neighboring cells, and as well as the 3D environment within the context of an intact tissue. So working together with Luo Lab, we designed the following strategy. We targeted HRP, which is another peroxidase that uses identical labeling chemistry, just like APEX that I mentioned earlier, to the cell surface of projection neurons with the cell type specific GAL4 driver. And then to further restrict the labeling chemistry to the cell surface, we used a biotin, a cell impermeable biotin containing small molecule probe. Following dissecting out the fly brain and applying hydrogen peroxide ex vivo, HRP is able to catalyze the biotinylation of cell surface proteins on projection neuron surface within a few minutes. So this fast temporal resolution really allows us to perform quantitative proteomic comparison of the cell surface proteome landscape at two critical developmental time points of the Drosophila olfactory system, which is the pupa stage, as well as the mature adult stage. So this workflow or example sort of illustrate one possible interpretation or recipe for 4D cellular physiology, which is performing molecular profiling, spatially resolved molecular profiling at biologically meaningful time points using physiologically relevant systems. in this case, an intact Drosophila brain. So our method was able to recapitulate many of the key findings, previous findings in the field. For example, we enrich protein complexes related to synaptic transmission in the adult stage versus protein complexes and molecules involved in circuit wiring and neuronal development in the pupa stage. More importantly, this unbiased quantitative comparison between the mature and developing stages allows us to further identify some of the previously unknown genes that could play an essential role in mediating neuronal circuit wiring process. And we performed an in vivo screen of some of the hits that we identified, and we achieved a very high precision in terms of validating the hits that we identified. And more importantly, a lot of the hits we identified came from unexpected molecular families, such as iron transporters and peptidase that have not been associated with neuronal wiring before. So this really highlights the power of performing in situ unbiased proteomic profiling in terms of expanding the molecular searching space to identify novel molecules related to neuronal wiring. So I want to use this final slide to highlight the potential of in situ spatial proteomics approaches for 4D cellular physiology, and especially to reveal the heterogeneity in terms of cell-cell interaction and cell environment interaction within the context of tissue. And while spatial transcriptomics and single cell approaches have been absolutely transformative in terms of expanding our understanding of this tissue level heterogeneity, if you're only looking at things at the RNA level, a lot of really essential elements are still missing. For example, there are circulating systemic factors that are produced by distal organs and then carried out through blood vessels into your organ of interest. And those will not be captured by transcriptomic analysis. And there are also factors derived from tissue innervating nerves whose cell body are located in ganglia that are outside of the tissue. And then finally, they're going from RNA to protein, there's tons of post-transcriptional regulation events that might shape the final protein product. So all of these factors play an essential role if we want to fully understand a range of biological questions, such as how the brain communicates with different organs, or what is the composition of tumor microenvironment or stem cell niche. So to really develop new tools for in situ spatial proteomics, it really calls for the development of novel molecular tools, starting from new labeling chemistry and enrichment chemistry, and also engineering novel enzymes and probes that are more compatible with using it in a tissue setting. So with that, I'd like to acknowledge my advisors, especially Alice, and also key collaborators at Stanford and elsewhere, and also my funding source for postdoc and graduate work. Thank you. Happy to take any questions.
Thank you so much for a really nice talk. Could you go back to your last slide, actually? The one where you talked about your limitations. I was wondering if you could expand on some of those development of new molecular tools. If you could expand on what you think those might be, what exactly, like what kind of enzymes do you think are going to help penetrate the different types of tissues, things like that?
Yeah, so that's a great question, and obviously an important direction moving forward. For example, in the peroxidase enzyme that I talked about, one of the things that's really limiting in terms of using these in tissue context is that you have to deliver both a small molecule probe and also hydrogen peroxide, which can be toxic to tissue if you incubate with a very long period of time. So I think that really so to develop new tools, it really comes from two angles. First is to use reagents that are less toxic. so that you can incubate or with tissue for longer periods of time to increase penetration. And then second is to use molecules that are chemically more permeable and easily dissolved and penetrate deeper into the tissue. Yeah.
We have a hand from Luke.
Yeah, I just want to follow up on that. Is the best way then to re-engineer make a better biotin thing, and like make it super permeable? Or do you feel a better way is to find a really bioavailable small molecule or even an endogenous molecule and then try and build an enzyme around that? What do you think is the best way to go after that problem?
I think the most ideal way would be to circumvent biotin altogether, because there are issues associated with biotin, because in mammalian tissues, there are endogenously biotinylated proteins. There are 5 mitochondrial proteins that are that have endogenous biotin modification. So when you're working with a very tiny amount of tissue, or you're trying to enrich a tiny amount of material, those endogenously biotinylated material really becomes a problem, and that increases your background and also limits the amount of material you can get. So if we can invent a pair that has similar affinity to biotin streptavidin, but also is completely bioorthogonal, I think that and easy to deliver, I think that would be the most ideal. Most ideal. Yeah.
So then I guess in your last minute, could you also comment the same way about like what type of labeling and enrichment chemistry would solve the problem that you're describing?
Yeah, in terms of labeling, I think ideally it would be fast, so that it has very fast temporal resolution, similar to Apex, because for proximity labeling, there are other tools such as biotin ligase, bio-ID based methods. So one major limitation for those class of methods is that the labeling takes a really long time. You need to accumulate material over 24 hours to get enough material. So that wouldn't be feasible if you want to look at, for example, the developmental time points that we're studying in the Drosophila brain development. So I think temporal resolution is really a key issue with some of these tools. And that can be overcome by through directed evolution of some of those enzymes. So Alison's lab recently engineered a new version of bio-ID called turbo-ID that is much faster. I think that's a promising direction going forward, just to take some of the enzyme that's already available in nature and then use directed evolution to make them with faster turnover and more promiscuous substrate. Yeah, so I think that's one cool direction to move forward with.
All right, thank you so much for a great talk. I think we're going a little over in time. There's another question for you in the Q&A if you want to answer it on the side. But okay, now I think we're moving into our discussion. So I'm going to be joined by Zhuoran Meier and Raj Chari.
All right. I think maybe I'll go ahead and get started with a question that kind of pertains to maybe one of the talks, but I think has some maybe broader conceptual questions. So this kind of goes towards Shraga. So I thought one thing that was really interesting, right, is that many of us who have thought a little bit about RNA modifications have heard a lot about M6A, and it's been the subject of endless Science Cell and Nature papers. And I thought you encapsulated nicely how it's a little bit maybe frustrating or difficult to say exactly what it does still, despite all that work, which is all really good work. And so one question I had is kind of to what extent do you think the fact that M6A is tied to this many phenotypes, and it's been tied to those via molecular profiling, pull down and sequencing of the modification, right? And those genomics approaches are powerful, but they also have limitations in terms of resolution. There was a recent study from Chris Mason and Stacy Horner where they kind of talked about the extent to which many antibody-based pull down studies of M6A are underpowered. And so I guess the question is, kind of one, there's been a lot of talk about technology and profiling technologies and big data sets. And, to what extent, can those correlation, sometimes, be responsible for leading you to think something's involved in lots of different areas, because you're correlating one big data set that may be low resolution in terms of its base resolution, in the case of M6A often, with RNA-seq data, with splicing data, with every different kind of ribosome profiling, molecular profiling data you can generate. And I guess as a larger, question maybe to the four of you, how have these different fields, be it single cell sequencing, long read sequencing, how are they converging on sort of established standards, and could there be a role for Janelia, HHMI, these kind of discussions in the community about that? So that's a big question. Shroff, you can start a little bit maybe with pass it around.
If I understood the question correctly, you're asking to what extent do limitations in quantification and in resolution of M6A, to what extent are they responsible kind of for potential confusion about what the roles of M6A might be? Is that the proper rephrasing of the question, or?
Much more succinct encapsulation.
Okay. So it's obviously something we've been spending quite a lot of thought on, and I'll say two things. So the resolution, at least of antibody-based methods, at least the initial ones that we had developed initially, we were talking about a resolution of, let's say, 50 to 100 bases, because it was based on capturing of short fragments with the antibody. But quite rapidly, work already in the field by different groups, and then also by later on also by ourselves, we were able to really increase this resolution down to the single base resolution, so where we could say precisely what site is modified. But what I would argue is that this resolution is not enough, and it goes a bit to the question that was asked before about M6As and how they matter and how they should actually be quantified. And I actually think the main difficulty is that when you have such an abundant modification, so you have this modification, and you can get better and better in terms of quantifying the stoichiometry of the modification at an individual site, which is one resolution to look at it. But then you're trying to correlate it against things that happen, processes that happen not at the level of the site, but that happen at the level of the gene. So the question is, how do you define a metric? How do you capture the overall M6A-ness of a gene? So how much M6A it has overall? And I think if there's one thing that's been challenging the field, it's exactly that, the lack of a measure that allows us to go from M6 at a site level to M6A at a gene level. And that's one of those insights that we had in recent work of ours, which will hopefully come out soon, is precisely that. So we were able to come up with this metric that integrates the overall amount of M6A across an entire gene. And suddenly when you try to relate it to different data sets that we've assembled, both in localization and stability and so forth, suddenly things fall into place, and you start seeing agreements where before we start seeing strong correlations, but where before you were seeing kind of weak effects. So I think it is a question of resolution, but it's not only the single site, the single nucleotide resolution we're talking about. It's more about integrating multiple events happening across multiple transcripts at multiple distinct sites, right? Because one transcript might be methylated at position A and B, and another one could be of the same gene could be methylated at the different positions, exactly because everything is substoichiometric. And then you somehow have to find a way to integrate all of this together.
Okay, so that's a very M6A, specific answer, and it's super, illuminating and interesting to think about. I guess, the broader question is kind of, I guess, as you kind of talked about these methods developing and progressing, right, in M6A field, right, from maybe a lower resolution antibody to more high resolution type methods now, so, I guess how, how does the field sort of move along, with establishing standards, and supplanting data, I guess, is kind of part of the question. Yeah, I'll just kind of open it up. Any thoughts that sort of, putting maybe Aaron or Winston?
Yeah, no. I mean, this is a really good question. Like, let me try to address it just from the context of like human cell atlas, just for lack of a better framework. So like, as single cell RNA sequencing has become standardized through, tools like the Droplet 10x genomics platform, the data starts to look more and more similar. And so people get used to seeing these, U maps of maps where you have cells and they're basically organized by their gene expression data. And now you can ask a question like, well, how do you overlay something like surface protein information on top of that? And I'm using this as an example because this is a little bit about what we do, but I'm going to try to generalize in a second. So one way you could do it is you could take your reduced dimensional gene expression space and start to plot protein levels onto it. But the more and more proteins you get, the more and more harder it is to do that one by one. And so, it'd be nice to integrate that protein data into the same lower dimensional space that you're using, that you're organizing cells with gene expression. And what that requires first is you to understand the differences in the measurement of the proteins in the RNA, because you can't just concatenate the matrix because you're going to have all these different biases. And so, what we do is we model the protein data and we model the RNA data, and then we can put it into a lower dimensional latent space. But then how do you layer on epigenomic information, right? And well, the first thing is you need, the throughput that on the single cell level. And so like people have started to do this with like a TAC-seq. But now you have to think about all the sparsity and the noise in the TAC-seq. And so like for every time you layer on another layer of measurement, you have to consider, all of the technical systematic aspects with that measurement. So then you ask, well, how do you layer on like imaging data? Like, I don't know. That's what we're trying to figure out. And we don't have the throughput like we were talking about. And then how do you layer on long read data? Which is a totally different format. Like, I don't know. I think it's easy to think about it conceptually, but maybe a better question is then, well, how do you use human cell atlas information in another space? Like, how do you just take cell type annotation from a lower dimensional cell atlas, throw out all your UMAPs, and then just use that sort of cell type list with the genes that are corresponding to that cell type and now map it onto a different experiment? Like, you have to think about, because I think right now it's sort of bespoke for every multimodal measurement that anybody does in their lab. They do their own data processing and manipulation and look at it in their own way. And I mean, I think that a generalized standard might be a little bit far off, but I think you can start thinking about just the way that information is organized in each one of these different measurements.
I would just to speak up, I would agree with that. I think that, oftentimes, part of the argument I was making in my talk was that we want to layer everything on the same molecule, so at least the correlation is known. But I still think it's really fair that you can't always do that. And there's lots of measurements where we're guessing about how the interaction goes, and you have these sparse matrices, and you have these you're adding dimensionality, and then figuring out what matters is hard. We usually try and reduce it by cheating. We say, oh, I just want to look at this subset of genes I already know is important, or these genes where I have differential expression, do any of them also have changes in M6A, or do any of them also have changes in the epigenome flanking them? Because it is sort of de novo discovery from this rich, massive data set becomes just intractable. The space is too big to explore, at least with current conventional tools. I think there's a need for tools that can integrate and handle this. And I also argue that there's speaking of 10x, there's new 10x sort of combined ATAC-seq single cell. So then they know from the same cell, again, this is sort of talking about getting the same information and coding in that information belongs to the same unit. That helps, I think. And so talking to Shroff's point also, knowing that where M6A is on the same gene, not necessarily the same A, but that this gene is heavily M6A, or this 3' UTR is heavily M6A, and another is not. Integrating it in each unit, and somehow then you can reduce the dimensionality, as Aaron was saying, to per unit, and splitting it up that way. I guess that's what I would say.
Yeah, and if you think about now going to combining with the microscopy workshop that we had earlier, So then, okay, let's say you can image, high density protein information, or you could do some in situ sequencing to get spatial information. You may not be able to do genome or proteome wide in situ sequencing, but maybe can you use some of these other data sets to kind of, to map a gene expression or a protein, composition identity into your images, which is a hard computational problem, but people are working on that.
I've seen that actually, Aaron, with like the 10x Visium stuff. You first cluster, and then you color on the spatial on the microscopy image from the same Visium slide the cluster that the cells have.
Yeah, exactly. And so, kind of patchworking our understanding of 4D physiology by using high resolution here, high dynamic range there. But I still think it is kind of ad hoc, and not in a bad way, but just because the data is so different,
When you're off the edge, it's always ad hoc.
Yeah.
I think Raj and Sarah, you guys.
Yeah, I do have one question. And it's sort of related to this talking about multi-omics and layering all this different information. How do you sort of look at the view of the trade-off of like signal to noise, right? And you both had touched on it in terms of this, adding these sparse data and adding all these different measurements that you're looking for. It all sounds very grandiose, like Shragi's slide, the dream of having everything and looking at everything. But there's also a practical and a realistic type of view at that. So in terms of, and this was open to any of the speakers, is that, how much, how do you see that sort of trade-off working? And also, how much data do you typically throw out that doesn't meet the quality that you're looking for? Because, I know a lot of people have experienced, especially with single cell RNA sequencing, as well as doing the combined, single cell with APAC or single cell with CRISPR, you do get a lot of kind of crappy data that you can't really use from. And so the whole answer to everything seems to be just sequence it way more so that you have enough good data and just, throw out whatever you don't need. So I would love some comments on that and thoughts on that. And I'll leave it to whoever wants to take that one. So,
I mean, I'll say really quickly that it's the tripod, right? You can be cheap, you can be accurate, or you can, what's the third one?
Fast.
Or you can be fast, and you have to pick two. And so, like, you can spend a lot of money sequencing deeply to do to your single cell point, or you can have really accurate, but maybe it's way more expensive and it takes longer. It depends a lot on and then the last thing I'll say, and then I'll give it to the other two. It depends on your application. Like ATAC-seq, let's say the sequencing, I'm looking at the sequencing read accuracy. I don't care. I just need it to map somewhere in the genome. And so there could be lots of errors in it, and it's okay. M6A, Shragi's arguing that we can do it per gene. And so, I mean, he should say, but maybe then I don't need per base resolution. As sort of Zhuoran was getting at, that might not be that great resolution. Who cares if I just care about the gene level? So I think it depends a lot on your application and what your question is, what the best answer to that is, and how much you care about signal and noise. If I care about like specific SNPs, I care a lot about signal noise. If I don't, then maybe I don't care. I just need a map.
Yeah, Arjun Raj asked a good question on Twitter, which was, would you prefer less better data or more worse data, essentially? And like, I think the response was like 50-50. I think it's a really good question because it's kind of like, which camp are you coming from? For the single cell stuff, we used to, and we still do like a lot, like trap one cell, high resolution imaging, sequence super deep. It takes a long time, but if we're asking specific questions, we can get really good answers. But then, with tools like variational autoencoders, more samples at sparser sampling can be extremely powerful. And like we could show that with these site-seq measurements where, more cells actually gives you a lot of power to fill in the gaps, given that some of your measurement techniques are sparse anyways. And, I think it does have to do a lot with the question you're asking. If you're trying to map the space of your system, you probably want more samples. But if you're trying to like understand a mechanism, you probably want better data. And, there's probably scales in between the two ends of the spectrum as well, like targeted questions and so forth. Like with the lamina, for example, that we were looking at, you can't really do FACS because you need the resolution to see these kind of curious laminar morphologies. And so even if you could kind of lower your resolution and increase your sample number, you might miss what you were looking for, which is also why people would do targeted sequencing as opposed to whole genome sequencing for specific questions, given that you have limited money.
Yeah, I think these answers also mirror a lot what we're seeing. It's really, really dependent on what we want to do and what we want to know. Often we use the same tools in a kind of surveyor kind of mode, and then we do things very shallowly, but it allows us to go at high throughput. And then if we're really interested in getting the maps right for in-depth follow-up questions, or just to get them to become as detailed as possible, then go a lot deeper. So I think Aaron and Winston really summarized the considerations really well also from our point of view.
There's something that Jordan and Raj and I were talking about when we were thinking about what we want to discuss during the session was we talk a lot about, when we're talking about omics, we talk a lot about how can we get more information and map more things and get all of the things on the same sequences. But what do you guys think it would take to do like a paradigm shift where we stop mapping and we shift over to like functional assays, like figure out, okay, if you had all of the information about every single sequence or every single protein that was interacting with it, what would it take to it to the next level and figure out what it's doing there?
Yeah, I think people who are better biologists than I am have been doing that kind of hypothesis-driven questioning for a really long time, and people are really good at it. Starting with a specific question, choosing your protein or your gene, and functional assays. Kind of coming into this field from an engineering point of view, I like to think of some of our tools as potentially just like the beginning of hypothesis generating. Like you said, mapping human cell atlas is a great resource. It's not an answer, to any specific mechanistic question you might have. Like in the case of, so we're doing a project where we're looking at the CD4 and CD8 T cell development from that common double positive progenitor in the thymus, and we can get surface proteins of hundreds of proteins and a genome-wide gene expression from these site-seq measurements, and we can map both lineages in pseudotime really well, and we get this really high density, high resolution map of this process, this decision-making process. But when it comes to asking questions like what happens before the branching point, this map only generates questions. Like it might be this transcription factor. And then we have to go in and ask, okay, if we inhibit that transcription factor or inhibit some of the signaling pathways upstream of that transcription factor in a model, does it in fact, perturb the amount of CD4 or CD8 T cells? And that's what, I'm learning a lot from, immunologists in that space, because there's a whole history of this hypothesis-driven questioning that people are really good at, obviously.
I agree. I think that you have to get it function, it's not enough just to characterize, you have to compare. And so you want to have sets of samples that are different in some way. If I'm ever trying to ask a functional question, it usually is because I have, being at Hopkins, I have case control from some human samples. And that allows me to say, okay, this actually matters because I have an N of 5 versus N of 5, and I see that difference is consistent between samples using some tool I've developed. But being a tool builder, I spend more time on, building the tool, and then I work with others, as Aaron's sort of saying, immunologist. I work with others to then apply it on hypotheses that they've generated. It's fun because I get to learn about the other areas, but it's not something I have probably thought about enough.
And Kaspar made a great point. Perturbation, right? This is not only just a classic tool in biology, but in physics. You have this theory of the system, and then if you want to see if this makes sense, you tweak it and see if you get an expected outcome or not. And so that's a really good point that you made on the chat.
All right, I think we might be out of time. Is that right, Jordan?
Yep. I think we're about just a minute over or so. So I think we'll wrap this session.
Yep. So. That sounds good. Thank you guys so much for a great session. And that actually concludes the program for today. So I want to thank all of the speakers and all of our discussion leaders for today. It was some really good topics were brought up, and hopefully we can continue to discuss them in our closed panelist discussion. So if the panelists could head over there, and you could take a break, please, for a few minutes and then join us in the discussion, that would be great. And as far as the audience, thanks for joining us today. And we hope you'll come back and join us tomorrow for another 3 sessions starting at 11 a.m. Eastern. So thanks, everyone. See you tomorrow.